All righty. Good afternoon. It is Tuesday, March 16th, 2021. We are on to the fourth section of the class. We've rapidly hit the halfway point and are continuing to moving forward. And we're going to be focusing on the muscular system. Way back when we talked about epithelial tissues and connective tissues, we also mentioned how there was muscle tissue. So we're going to start out by talking about the anatomy and physiology of that muscle tissue and putting that together and the histology associated with that. And we'll start that discussion today. And then we're going to spend the rest of this section basically like we did for the skeletal system, really separating the lab and lecture portion, where in the lab we'll be talking a lot about the physiology of muscle, and then in lecture we'll be focusing on the muscles themselves. Towards that goal, in understanding this material for Thursday, starting on Thursday's lecture, you will need a red pen and a blue pen. Sorry, there's a little bit of background noise. I'm going to go ahead and mute that for starters, and then you guys can unmute as needed. Either red or blue pens or pencils to do some drawing to go along with the handout that is on Canvas that you'll need for the origins and the insertions. We're going to be drawing the origins and insertions. The origins and insertions aren't new material. They're many of the bone and bone features that you learned in the last section. So we'll be using them again. And by convention, and remember, by convention just means a rule that a bunch of people got together and made. Red is the color that is used for origins, and blue is the color that is used for insertions. So we will continue that tradition. So make sure you have those so that you can draw them. Also on Thursday, following up on the muscle tissue stuff we're going to be doing today, your first labster is due. Remember, for full credit on those labsters, you have to complete the entire activity, and you must get it 80% correct. And again, remember, if you don't get it 80% correct, you do have the ability to go back and do it again. Your highest grade does count. So I know there's a couple people who have not gotten 80% correct, and if you don't redo it, you don't get full credit. So take the time to go through it again. Once you've done it once, it's much easier to do it a second time because you know all the tricks to it. And so complete that to get full credit for that. Also, before the break on Thursday, the 23rd, again, we do have class on Tuesday, the 23rd, Tuesday, the 23rd, but there's no assignments due on that day, although I will remind you that on a day when there's no assignments due, that's always a good day to have a daily quiz at the beginning of class. But on Thursday, the 25th, your first unit review is due, and that's unit review 10 that you'll be turning in those pages. And then we get spring break. And then again, to keep you busy during spring break, when we come back on Tuesday, the 6th, your PhysioX exercise 2 is due. I know many of you had a tremendously fun time with the first PhysioX, and so this one is just as much fun, if not even more so, because there are 7 activities, which means 7 lab reports. And again, don't print them out. You'd kill a small tree to do that. Save them as PDFs and submit them as PDFs to the Dropbox on Canvas. And those are due, remember, at the beginning of class. So that should keep you pretty busy over break, aside from all the other studying that you're going to be doing as well. All right. Questions on any of that? Excellent. I thoroughly enjoy my stunned silences first thing in the morning. So let's go ahead and get started then. Move that here and here, get that out of my way, and we will switch to lecture. All righty. So when we talked about tissues, we talked about some of the cardinal characteristics of those tissues. Epithelial tissues are cell dense, they're avascular, they're superficial. Connective tissues have a matrix, and that matrix determines its characteristics, right? And we talked a little bit about muscle tissue, so let's talk about muscle tissue now. What are some of the functions? What do muscle tissues do for us?
Help move.
They move the bone.
Okay, excellent. I like that. Movement of the bones of the body. They move our body through space. Absolutely. So that is definitely one of the types of movements they allow, right, when we're talking about movements. But we also need to be careful because if, for instance, I stand back here and I put my hands out and I hold my breath and close my eyes, is there any movement going on then? Are muscles doing anything if I'm doing that?
Yes, because your heart is pumping.
Excellent. So when we think of movement, another type of movement that our muscle tissue allows for us is the movement of substances through the hollow organs of our body. The heart definitely does that. Any other examples of that going on? Yep, we'll get to that one in a second. Anything else other than the heart? Digestive system, right? That cheeseburger I had for breakfast, stomach and the small intestine, all of those things are moving those things through those. And of course, as we know, the way you do that is these hollow organs change their volume, which, as we know, changes the pressure. And as we know, pressure is what makes the world go round. But you're right. Muscles are doing something else when I'm standing there with my eyes closed and my holding my breath. They're stabilizing my body in position. They're taking small little contractions that may not be visible necessarily, but allow us to hold ourselves in place. So that stabilization and locked in place balance. Right? We take this type of thing for granted until you've had a few too many Jägermeisters. Then suddenly your ability to be able to stabilize your body position and hold it in place goes away. Right? By the same token, right, when I'm not moving my right arm doesn't suddenly sag out of position where it is in being held by that glenoid cavity. So not only does it provide stabilization and balance of the body, but it also helps to support and hold the joints in place. What else does muscle tissue do for us?
Maintains body temperature?
Excellent. Absolutely. It plays a huge role in helping to regulate body temperature. How does that work again? Does anybody remember? Yeah, exactly. They produce a tremendous amount of heat. The reason they're able to do that is muscle involves motors. Right? What's a motor again? I think we've talked about this in this class, but I don't remember for 100% certain because usually I use the example of the cars that got you here to class today, but many of you probably haven't driven a car like me in days or even weeks. What does a motor do?
Changes the type of energy from chemical to mechanical, or the opposite.
Perfect. Yep, it converts chemical energy into mechanical energy. Right? Absolutely. That is exactly what it does. And like most muscles, like most motors, I should say, our muscles are not 100% efficient. In fact, some of our muscles, as much as 75% of the energy that is converted from chemical energy doesn't make it into mechanical energy. As much as 75% of that is instead lost as heat. Now, that isn't a very effective motor. You probably wouldn't want that engine in your car if only 25% or if 75% of your gasoline was lost as heat, right? That wouldn't be a very efficient way of doing it, but it plays a huge role in helping us to regulate our body temperature. Excellent, excellent, excellent. What else? I can think of one more really, or actually two more really important functions of our muscle tissue. How about this one? How does our muscle tissue provide protection?
Doesn't it help line like the cavities of where the organs are?
Yeah, absolutely, right? We think of up here, we have these bones. We have these bones that are protecting these organs like our heart, like our lung, like our brain. But what about all the organs in your abdominal pelvic cavity? Are most of those protected by bones?
No.
No. Instead, you have a muscular girdle that actually helps to hold and protect those in place. In fact, that's how Harry Houdini died. Harry Houdini, as many of you know, was an escape artist and magician. And again, back in the days when he would perform, it takes many of the escape things that he would do, like escaping from the milk can or escaping from other types of things, would take a bit of time to prep. So when he would put on a show, he would do one of his escape bits, and then they would have other people perform or things like that while they set up, and then he would do his next trick. Well, one of the things that they would do in these intervals between his big tricks is he would come out on stage and brag about his constitution, and he would invite anybody in the audience that wants to come up and punch him in the stomach. And the men would line up to come out and do that, and they'd punch him in the stomach as hard as they could, and he'd laugh at them and mock them, and the crowd would laugh and have a good old time with it, because he had this tremendous strength to his core. Well, one evening, a day or two after a show, he was coming into a bar as two college kids were coming out of the bar that had seen his show a couple nights before. And without any warning and without saying anything to them, they punched him in the stomach 2 or 3 times. He didn't have the opportunity to engage his core to protect himself, and they ended up rupturing his spleen, and he died a couple days later. All right, so our ability to, protect ourselves with that core is something that is vitally, vitally important. Does muscle do anything else for us?
I'm not sure if I'm correct, but does it help with like nutrients?
Absolutely. It is what we call a nutrient reserve. Right? Think of it this way. Summer is right around the corner. With summer right around the corner, we all have to get into our bikini bodies. I know I do. Right? I've been putting on that COVID-10 that they keep talking about. And so I decided to go on the popcorn diet. Right? And that's where I have 1 piece of popcorn for breakfast, 1 piece of popcorn for lunch, and a generous 3 pieces of popcorn for dinner. Right? If that is my diet, am I going to lose weight as a result of that?
No.
Yeah, I am, because I'm going to be starving myself. However, the key is, am I going to be losing fat when I do that? Is the fat just going to be melting out when I'm doing that?
No, you're gonna lose muscle as well.
Exactly, right? Fat is actually a very valuable resource in your body. When you put your body into starvation mode, your body actually holds on to the fat. It wants to protect that fat because that is a valuable resource. So instead, as you pointed out, what gets broken down to provide the resources that you need is your muscle. You actually break down muscle. Muscle is just chock filled with massive amounts of proteins. And so if your body goes into starvation mode, it actually is able to break down those proteins inside of the muscles to provide resources for your body to be able to survive, right? Our goal under normal circumstances, normal healthy circumstances, yes, typically we go carbs, fat, and then proteins in that way. However, if you are putting your body into starvation mode, that actually changes. Right, as you're starving the body, then it isn't going to work in that fashion. Yes, Laura, did you have a question?
Yeah, so is it like kind of the opposite? So if you're a healthy person and you're trying to um lose fat and gain muscle, is it like the opposite? Do you I guess what I'm trying to say is so you lose your
So,
muscle first when you're starving yourself, and then when you're trying to get it back, that's the last thing you get is the muscle. That makes sense.
I understand what you're saying. Not necessarily. Here's what I would say. So yes, clearly when you put your body into starvation mode, then the problem is, yes, it is much more likely to break down muscle, to metabolize that muscle to be able to provide the nutrients and the resources that you need. So the most effective way to lose the weight, or for that matter gain muscle, is basically exercise. There are many things, if you think back to biology, when we were talking about, well, not we, but when you were taking a general biology class, and they were talking about things like Mendelian genetics, and one of the opponents to that was a gentleman by the name of Lamarck. He was the guy who thought that the giraffes necks got longer because they used them. The more they used them to reach those taller leaves, stretched them out, and they came up with this concept of use it or lose it kind of thing. And muscle truly is the use it or lose it type of tissue in our body. The more you use the muscle, the more active it's going to be, the more you're going to build proteins into those muscles and maintain them. So the best way to lose weight and maintain muscle mass is to, yes, restrict your caloric intake, but also to make sure that you're active enough to maintain the muscle tissue while you're doing that.
Gotcha.
Yes. Yeah, absolutely. There you go. So we'll get into some of that more when you get to digestion, but the short answer of a keto diet is it puts the goal is to put your body into a state of ketoacidosis where you are more likely to metabolize fats than you are carbohydrates. So basically, it's again about a change in the activity levels, a change in the amounts of nutrients that are available to try to encourage the body to metabolize fats rather than carbohydrates. So that's basically how that works. All right. Excellent. So I think we've hit all the big stuff here. So we can look at what I've got here, right? Producing body movements, stabilizing joint position, moving substances within the body, supporting the soft tissues in the joints, maintaining body temperature, nutrient reserve. Excellent. We've got all of that. Yeah, it can't. You're right. It is. And like I said, it is something worth debating, although it's not something that I think we necessarily need to discuss in there. When we get to the digestive system in 431, that'd probably be a good time to talk, because that's when we're going to talk about nutrition as well. So that would be probably a good time to talk about that then. In simplest terms, yes. Lowering your caloric intake, having more output of energy than you have input of energy, and maintaining muscle mass. Those are the basics. But again, all of these things are a little bit more complicated. You could have two people of the exact same size, the exact same weight, exact same muscle mass, who ingest the exact same amount of food, do the exact same amount of activity, and yet their weight gain or losses are going to be different. So it is a little bit more complicated than that. Kind of like everything in biology, right? Everything in biology is a little bit more complicated than it seems. All right. So we know now what muscle tissue does for us. And as we talked about way back in the day, there are three main types of muscle tissue. What are the three main types of muscle tissue again?
Skeletal, cardiac.
And
Smooth.
smooth. There you go. Cardiac, skeletal, and smooth. Absolutely. And just like there are 8 different types of epithelial tissues, right? But those epithelial tissues all share certain characteristics. The same thing is true for all muscle tissues. All muscle tissues, smooth, cardiac, skeletal, all share 4 main characteristics or 4 main properties, right? The first of these is that muscle tissue is irritable. What does it mean to be irritable?
Response to stimuli?
Yeah, and actually, exactly. That is exactly what it means to be irritable, right? My favorite example of this is my grandmother. My grandmother is the most loving, most caring, most sweetest individual in the world, except between noon and 1 o'clock Monday through Friday. Because between noon and 1 o'clock on Monday through Friday is when grandma's stories are on. And you do not interrupt grandma's stories, right? If you talk during grandma's stories, right, she will take off her shoe and throw it at you, right? And that's what it means to be irritable. You receive a stimulus and respond to it, which I think it says there. You receive a stimulus and you respond to that stimulus. That's what it means to be irritable. Now, in the case of all of our muscle tissues, these muscle tissues respond to being stimulated by producing a big positive electrical signal. Anybody know the fancy term we give for this big positive electrical signal? Say again.
Action potential?
Action potential. Right. So muscle tissue, when is excited, and actually let's do it this way, produces what we call a muscle action potential, a big, huge positive signal. And so since it produces a big, huge electrical signal, we can also say that it is electrically excitable. When it is irritated, it produces an electrical signal, a muscle action potential. Now, that muscle action potential causes the muscle cell to contract. And that contractility is the second major characteristic or second major property of our muscle tissue. And what does it mean to have contractility?
To get shorter or smaller.
Excellent. The cell changes shape. And as it changes shape, basically it generates a force. It generates a tension. Right? That produces a movement. Right? This generation of force, this generation of tension, this contractility, right? That allows it to shorten. And when it is shortened, right? When it's stimulated. Now, notice both of these are what we could consider active properties. What do I mean by that?
It's always going on, like there's no, or I guess is it like involuntary?
No, I guess some of it is involuntary. Obviously, when I have that cheeseburger for breakfast, I don't have to tell my stomach to start churning, right? However, if I want to wave to you guys on camera, obviously that is something that is voluntary. So it isn't necessarily about voluntary or involuntary. What does it mean to be active? When we've talked about using terms like active in the past, what does that mean?
It requires energy.
Yeah, it requires energy, primarily in the form of ATP, right? This is something that the muscle tissue does. Uses ATP to do these activities. It uses energy to do these activities. This is important because, as we will see, the second two properties of our muscle tissue are actually what we would call passive properties. Oh, I don't have one, did I? I didn't prepare ahead of time. Nope, I didn't. All right. The third property, the first passive property, is extensibility. What does extensibility mean?
Extend or stretch?
Okay, you absolutely. However, if you think about it, if I had some saltwater taffy here, I could stretch that as well. Would that have necessarily extensibility in the same way?
No, or I guess and extend it to a point where it doesn't get damaged.
Absolutely. That's what it means, extensibility means. And that's what I was looking for. I was looking for a rubber band. Notice if I had a rubber band, and we can all pretend I had a rubber band, I could stretch that rubber band out and it wouldn't be changed. It wouldn't be damaged by that process. That is what extensibility means, an ability to be stretched without damage. Now, like everything, there are limits. Can I stretch a rubber band to a point where it snaps?
Yeah.
Yeah, we talked about how skin has extensibility. It can be stretched out as well. However, as I also mentioned, if you become pregnant and your belly extends rapidly, or like I talked about with my brother-in-law who grew a full foot during his senior year in high school, he stretched his skin beyond its extensibility, and he has stretch marks as a result of that. So there are going to be limits, but it has that ability of the rubber band to be stretched out. And that's the key, right? It is to be stretched out. This is a passive process. If I take that rubber band and I put it here on the counter and I stare at it for the next 3 years, is it ever going to stretch itself out on its own?
No.
No. right? It can be stretched without damage, but it doesn't stretch itself out, right? And so this is a passive property. This is something that occurs to it, but it can't do itself, which again, if you think about it, makes sense. Because if I'm going to flex my elbow, I contract this muscle gets shorter, and it pulls my forearm up. If I decide I want to extend my elbow, does this muscle up here on top push against the arm, pushing it away, getting bigger as a result of that?
No.
No. So then how do I extend my elbow?
Your other muscles on the other side.
There you go. I contract these muscles on this side, which get shorter actively. And notice, while they actively get shorter, this muscle here is stretched out passively without being damaged. All right, so this is a passive property. It can be stretched out without damage.
So can you
Oh, go ahead.
I'm sorry. So can you kind of think of it like as the bladder, like it doesn't stretch until there's fluid in there?
Yeah. The bladder is an excellent example. The bladder also stretches passively. The bladder itself doesn't physically get bigger, right? It doesn't enlarge itself. It is stretched by the fluid inside of it. Absolutely. It's a great example. And like that rubber band, when I stretch it out and then I let go, what happens to it?
Goes back to its original shape.
Back to its original shape, right? And that property we call elasticity. The ability to, when you crumple up it into a ball and let it go, it goes back to its original shape. You stretch it out, it goes back to its original shape. And the same thing is true for our muscles as well. You stretch the muscle out, you contract the muscle down, you relax it, and it's going to go back to its normal length. And I will tell you right now that most muscles are at their normal length and shape when you are in anatomical position. So when you're in an anatomical position, most, not all, but most of your muscles are at their normal length and shape. All right. So again, most of the examples I just used involved skeletal muscle because it's the one that's the easiest to think about. But this is all four of these are also true for smooth muscle and also true for cardiac muscle. Yep, elasticity is also a passive property. That is correct. So most muscles, at anatomical position, and absolutely, this is also a passive property. Right, it's not using energy to go back to its original shape. So yeah, so 1 and 2 are active properties, use ATP. 3 and 4 are passive properties. They occur to the tissue, but it does not require ATP to do it. Excellent. And again, these are characteristics of all of our muscle tissues. All right, just like we learned characteristics of all of our epithelial tissues. All right, questions on that? All right, perfect. Let's get rid of that. The example of what happens to my bicep brachia, the bladder too. Bladder was a good example, right? The smooth muscle of the bladder is stretched by the urine inside of it. And notice when the urine leaves, it goes back to its original shape. So that's a great example of both extensibility and elasticity of our bladder. The example I actually used was in my bicep brachia. Remember, I contracted it. I actively used energy to flex my arm. But when I extend my arm, I use my tricep brachia. And as I'm using my tricep brachia, my bicep brachia is being extended. It is being stretched without damage by the energy of my tricep brachia. So those are the two examples that we talked about. Absolutely. All right. Great question. Any others on these 4 properties of all of our muscle tissues? All right. Well, then let's talk about our 3 types of muscle tissue. I think we've already mentioned them a couple times, but let's talk about some of the characteristics, some of the properties of them. And it's always fun to use the whiteboard for this, so let's go ahead and do that. Oops. There we go. All right, someone remind me again what the three flavors of muscle tissue are?
Skeletal, smooth, and cardiac.
Skeletal, smooth, and cardiac. Excellent. Where do we find skeletal muscle?
Attached to like the bones or in the facial.
Absolutely. Most skeletal muscle is attached to bones. Hence the name. Of course, what's the key word in that sentence?
Most.
Most, because most means not all. There are some, especially like facial muscles, that actually can attach muscle to muscle, or may actually attach muscle to skin or bone to skin, right? So that we can move our face around. So, but most skeletal muscle is attached to the bones. Excellent. Skeletal muscle is our largest of our cell types, has the largest cells. Some skeletal muscle cells can actually be a foot long. They also have a cylinder shape. So if we were to attempt to draw this, we would have a circle like that. Yes, Laura.
Sorry. So this is the part where I kind of got confused on the pre-lab. So it says cylinder shape. Does it necessarily mean that they're columnar cells, or is it just like the shape that they make is the cylinder? I don't know if that makes sense.
I'm not sure I understand. I don't remember the part of the pre-lab that you're referring to, but typically these cells are going to be long, thin, narrow cells.
Oh yeah, there was like a table at the last on the last page.
So pre-lab, number 10.
On page 294.
294. So what's the question?
So you mentioned that they're cylinder shaped. I put also cylinder shaped, but I guess my question to that is are they all columnar cells, or is that just they made?
I see what you're saying. Okay, I misunderstood. Yes. No, remember, columnar cells is the shape of the cells of epithelial tissues.
Oh, okay.
So these are going to, you could, like columnar cells, they are long and narrow.
Okay.
But the internal anatomy of them is incredibly different. And we're actually going to spend a tremendous amount of time talking about the specific anatomy of this. So great question, Yulia. Absolutely. That is another important characteristic of our skeletal cell. It is multinucleated. Which of course means that it has multiple nuclei. So let's go ahead and give it multiple nuclei. But even though it has multiple nuclei, it is still going to be considered one cell. Basically by the plasma membrane. Is how we can tell one from the other. And we'll look at this histologically and be able to distinguish this. So we will see this when we look at this histologically. After we're done writing this, we'll write it with pretty pictures. Now, what's the advantage of having multiple nuclei?
You're able to make proteins faster.
Absolutely. Remember, nuclei contain the information on the genes on how to make proteins. And it turns out skeletal muscle, all muscle, but skeletal muscle in particular, is just absolutely chock filled with these massively precisely arranged proteins. so much so that the nuclei in them are actually pushed out to the side. Obviously, they're still inside the plasma membrane, right? The plasma membrane of this cell still wraps around the entire cell. So the plasma membrane wraps around all the proteins, it wraps around the nuclei. Again, they're not pushed out of the cell, they're still in the cell, but they're pushed way to the side in the periphery. If you think about it, kind of the same way, remember our adipose cells have that big, huge fat vacuole that pushes the nucleus way out to the side. And basically the same thing happens here as well. All right. There's another important characteristic of skeletal muscle, and it is that it is striated. What does that mean?
Means it has like a stripe appearance to it.
Absolutely. Those proteins that are filling up this cell have such a precise arrangement to them that when you look at the inside of them, there's going to be some areas that have a large amount of dense dark proteins, and the next area is going to have less proteins, and then more precisely arranged dense proteins. And these proteins become so precisely arranged that they actually form a physical stripe to this cell that actually can be seen with a light microscope, right? If with a light microscope, we can't see individual proteins inside of the cell, but these proteins are so perfectly aligned that with a light microscope, we can actually see the areas that have more of them or denser of them and the areas that are not as dense. And so we can see those that striped appearance to that skeletal muscle cell. Excellent. All right, let's talk about what else we know about these cells. When we talk about their activity, is it something we consciously control or is it unconsciously controlled?
Conscious.
Just
All right. Voluntary control. And that voluntary, control is by a specific branch of our nervous system called the somatic, because soma means body. Mode. So actually, let's say it this way. Voluntary control, and it's controlled by our somatic motor nervous system. We know one other thing about that. If I want to wave my right hand, where do I actually make that decision?
The left side of your brain.
Yeah. Right. The left side of the brain is where we actually make that decision. What do we call that when the signal has to cross the midline? Anybody remember?
Cross-sectional? No?
Close.
Contralateral.
Contralateral. There you go. Contralateral. Absolutely. Contralateral. We have contralateral voluntary control of our skeletal muscle, right? Meaning that we voluntarily control it, and it's controlled by a branch of our nervous system called the somatic motor nervous system. Last characteristic we want to focus on is regeneration, right? We know when it comes to healing, there are two ways that our tissues heal, right? Regeneration or fibrosis. Regeneration is where we replace damaged tissue with the same type of tissue, maintaining the look, the feel, the function. Whereas remember, fibrosis is where we replace the tissue with a fibrous connective tissue that doesn't function the same way. And so when it comes to healing, does skeletal muscle regenerate?
Yes.
Yeah, if I pull a muscle working out today, I'm able to heal that muscle and replace that muscle with more skeletal muscle. However, if a shark takes my entire shoulder away, can I replace all of that skeletal muscle with regeneration?
No.
No. So it has a very limited regeneration. It has some regeneration, but that regeneration is very limited. Minor injuries can be healed via regeneration, but otherwise, larger injuries, fibrosis, and that's going to affect the feel, the look, and the function of that tissue. All right, Let's do the same thing for smooth muscle. Where is smooth muscle located?
The hollow organs.
Excellent. So again, most lines the walls of hollow organs. Of course, there's that pesky word again. What's that pesky word again?
Most.
Most. And most, of course, means not all. Can someone give me an example of smooth muscle that isn't lining the wall of a hollow organ?
Aren't your eyes made of smooth muscle?
So
Or
there
like
are
part of them?
portions, they're absolutely the iris that changes shape, the muscle that changes the focus. Those are definitely smooth muscle, absolutely, and that's 100% correct. I was actually thinking of something we already learned about in this class. Oh, there you go. Aravek's got it. The erector pili muscle, right? That was another example of something that we talked about there. Smooth muscle is our smallest cells, typically, and they are what we call spindle shape. A spindle shape basically means that it has a large centrally located body and then two tapered tails that come out. Obviously, this is not drawn to scale. It's not the same size as the cylinder. Arteries do have smooth muscle that line them, but again, remember, an artery is a hollow organ that contains blood inside of it. So that would still be, yeah, so digestive system, blood vessels, airways, most of the hollow organs in our body, all of the hollow organs of our body have smooth muscle, right? The uterus, we were talking about, the bladder, all of those have smooth muscle in them that have this spindle shape. With this spindle shape, they typically have a single centrally located nucleus. So we have, it's uninucleated, and it's a central nucleus. Right. These also, while they're spindle shaped, like we said, they line the hollow organs, so they're typically arranged in layers, interwoven with each other to form a layer of smooth muscle that lines those cavities. So we can put that there. None of which really explains why it's called smooth muscle. Why is it called smooth muscle?
Because movement?
Movement would be a good guess. It does have a very smooth, slower motion to its contraction. So that
Mainly
is
because anatomists hate us and skeletal muscle looks smoother.
Non-striated?
yeah, exactly. The reason it's actually called smooth is because it doesn't have the stripes that skeletal muscle has. So it is actually non-striated. Does not have stripes. Now, as we know, those stripes are caused by proteins, and those proteins are actually going to allow that skeletal muscle to contract. So because these don't have the stripes, they are actually going to use a very different mechanism to contract from skeletal muscle because they are non-striated. So again, it's a difference in their appearance, in their structure, but that structure affects their function. Well, that's a great point that we're getting to next. When it comes to control of them, again, is this something that you voluntarily control? Can you make your erector pili muscle stand up on end? Do you have to tell your stomach to contract? No, this is
They're involuntary.
Oops, involuntary control. Absolutely. And it is controlled by hormones. Absolutely, hormones can tell our smooth muscle what to do. But also our nervous system can as well. Now, it wouldn't be our somatic motor nervous system. Does anybody know the branch of the nervous system that would control our smooth muscle?
Axons.
Close.
Episomatic.
So We have somatic motor, which is our body, and then we also have our autonomic, right? Our autonomic nervous system. Our autonomic nervous system has two branches, sympathetic and parasympathetic. You may or may not have heard those terms before, but there are some cutesy little rhymes they use for those. What's the cutesy little rhyme for our sympathetic nervous system? Really? No one?
I don't know.
Sympathetic nervous system is what lets you deal with that bear with an axe that walks into the room. So when a bear with an axe walks into the room, you have two choices. What are the two choices?
Oh,
Fight or flight.
Fight or flight.
You either have to fight that bear with an axe or you have to outrun somebody in the room with you. All right, so sympathetic is our fight and flight, whereas parasympathetic then is what?
The opposite.
Yep, what is the opposite one? The one that takes over on Thanksgiving.
Or flight. So
About rest and digest. No one's heard that one before? It's okay if you haven't heard that one before. That one's not actually that accurate, but we will talk. Well, the good news is we'll get to the autonomic nervous system in the next section. So we will get there. It's okay that you guys don't know that now, but we will get there. So the take home message, what we need to know now is that this is something we do not consciously control. There's an involuntary control, and it's basically our hormones and our autonomic nervous system that control it. All right. And when it comes to healing, as it turns out, our smooth muscle has the highest rate of regeneration. So of all of our three muscle types, smooth muscle is the one that is easiest to regenerate, easiest to repair with the same type of tissue. All right. Questions on that?
Yes, I can't remember if you said the eyeball is smooth muscle tissue or not.
So again, the entire eyeball is not lined with smooth muscle, but within the eyeball, the iris, the thing that controls how much light that comes in and out of your eye, that is smooth muscle. The part that changes the focus of your lens so that you can look at your screen that's right in front of you and read words, but you can also look across the room and take something in and be able to see it clearly across the room as well. That ability to change the shape of your lens also is smooth muscle. So absolutely,
But the blinking isn't controlled by smooth muscle. Blinking is still skeletal muscle.
What isn't?
Blinking, like when you blink.
Yes. Blinking, yes. Blinking, moving your eyes through space, all of that is skeletal muscle. So moving your eye up, down, left, right, and blinking, yeah, all of that is skeletal muscle.
Okay, thank you.
Laura, yes, you had a question?
Yeah, just a quick one. I don't want to get ahead, but so you said on the skeletal, the reason it's striated is because it has those two proteins, and then
No,
the
I didn't say
smooth.
it has two proteins. It has a ton of proteins, but the proteins inside of them are very, very precisely arranged. And so there are some areas that are more dense with proteins and some areas that are less dense with proteins.
And then that, I don't want to get ahead, but that would be like the thicker the thin.
Yeah, they involve the thick and thin filaments, absolutely. And we'll actually talk about that today.
Thank you.
Yep, we will start seeing that today, absolutely. All right, spectacular. So yeah, it's this precisely arranged myofilaments, proteins inside of there, like the thick and thin filaments, absolutely. All right, excellent. Let's do cardiac muscle. What is the location of cardiac muscle?
The heart.
Now, mostly in the heart, like skeletal and smooth muscle?
Only in the heart.
Only in the heart, exactly. Cardiac muscle is found one place and one place only, and that is just in the heart. Now, obviously, if skeletal muscle is the largest and smooth muscle is the smallest, this one is going to be intermediate in size, in between the two. And it also has a unique shape. that unique shape is what we call a branching shape, or we can say that it is a branched shaped cell. What this means is that it is sort of cylindrical, but it can have tapered ends and branching ends to it. So again, they're not all going to look like these alligators like that, right? Some of them may have, you know, 2 branches on one side and 2 branches on the other, right? Or some could have 3 branches on one side and 2 on the other. They're going to have these branching pieces that come off of them. All right. And not only are they in between in size, but they kind of share some characteristics with smooth and skeletal. Like smooth muscle, they're uninucleated and have a centrally located nucleus. Yes, there is. Oh, you want to know what it is? My guess is that the reason, as we'll see, has to do with the way that cardiac muscle works together as a single unit. These cells, as we'll get to, have gap junctions that connect them to the cells next to them so that they all work together as a single unit, so that they function together in a uniform fashion. And my guess is the branching nature of them allow them to communicate with multiple cells, allowing the whole layer of cardiac muscle to work together as one. So has a single centrally located nucleus like smooth muscle, but like skeletal muscle, it is striated. It has stripes. And what this means is it has the same arrangement of proteins and the same type of proteins that we see in our skeletal muscle, which means it's going to contract in a very, very similar fashion to the way that our skeletal muscle contracts. So when we learn about skeletal muscle and how it contracts, we will see that cardiac muscle is going to function in a very, very similar fashion. The way it generates its tension, the way it changes its shape is very, very similar. Now, when it comes to control, is it voluntary or involuntary?
Involuntary.
Excellent. And let me ask you one other question. If I were to yank a skeletal muscle cell out of your body and put it in a petri dish and provide it with all the oxygen and all the nutrients that it needed and watched it for 3 years, would it ever change its shape on its own? Would it ever contract by itself?
No.
No, because as we said, it's controlled by the somatic motor nervous system. When I've yanked it out of your body, right, it no longer has that neural control and it can't be stimulated. So it's just going to sit there. It'll be alive, it'll be happy, but it's not going to contract. Same thing with the smooth muscle, right? If I put that smooth muscle in a petri dish and I provided it with oxygen and nutrients and I stared at it for 3 years, it would never change its shape either. But what about cardiac muscle? If I ripped your heart out of your chest, put it in a petri dish, and provided it with all the oxygen and the nutrients that it needed, would it contract on its own? Yeah. One of the special things about cardiac muscle is actually what we say autorhythmic. Autorhythmic basically means that it is capable of producing its own action potentials. Hold on 1 second, Sydney. I see your hand. Just give me 1 second. Yes, Sydney, what's your question?
So in the lab manual, it talks about cardiac typically having only 1 or 2 nuclei. Is that just for the sake of this class, we're going to go with uninucleated?
You are correct. There are the occasional cardiac cells that will have 2 nuclei, but there aren't enough of it where it's really significant. And again, even if it has 2, it's still far, far fewer than we see in the skeletal muscle, which will have dozens.
Okay, got it. Thanks.
Yulia, you had a question as well?
Yes, I was gonna ask you if you want to talk a little bit about heart transplants and tell us cool things about it.
I'm for them. How about that?
No, like the whole, like, does it keep beating when we switch it from one person to another, or like, how does that happen?
So, okay, great question. So, all right, let's go back to this idea of pulling the heart out of the chest. When we pull the heart out of the chest, the heart does have autorhythmicity where it's actually able to produce its own action potentials. In fact, if I pulled that heart out of your chest and put it in a petri dish and was able to provide it with all the oxygen and nutrients, it would beat at a rate of about 100 to 120 beats per minute. Is that what your heart is doing in your chest right now? Anybody's heart rate as there's, and unless you're exercising while we're doing this lecture, or maybe you had a quad shot from Dutch Brothers on before class, nobody's heart's probably beating 100 to 120 beats per minute right now. Right? So while it is autorhythmic, it is capable of producing its own action potentials. True, but nobody in this class is newborns. They would have had to have their waiver signed by their parents, and I would know if that occurred. So no, there's no newborns in this class, so those don't count either. But are there times when your heart beats that fast?
Yeah.
Yeah, absolutely. Or it can beat slower. So notice, while it's not controlled by our nervous system, by our hormones, it can be modified by our autonomic nervous system, right? That bear with an axe walks in your room, your heart rate's going to change, right? And hormones. So it can be modified that way. To answer your question, when someone has a heart transplant, the heart itself isn't necessarily beating because, again, it's been not frozen, but it's been cooled to a temperature where it's not very active. But the other problem is, again, you lack this ability to modify. So typically when you get a heart transplant, you are getting an artificial pacemaker to go along with it that will control it at the appropriate level, because you can't just rewire it into our nervous system in that fashion. So it doesn't work that way. So they do require artificial stimulation when those types of transplants occur.
Okay, cool. And how successful are heart transplants?
I don't have an answer for, I don't know the statistics on that. I know that compatibility is a major issue, right? When we talk about blood, even in a group as small as this, where there's 24 people here today, I guarantee you there are at least 2 people here where one person could give their blood to somebody else in this class, right? Because our cells have special proteins on the surface that allow our body to recognize them as being us or them as being not us. And we want to protect ourselves from things that are not us. With blood types, there's only really 2 proteins on the surfaces that we have to worry about. And even if you weren't aware of that, we definitely know that, because those people who know their blood type, it's something like O positive or A negative or something like that. It has 2 components to it. Things like the liver, things like the heart have over 200 proteins that can cause a severe reaction. And so being able to find a match that is perfect for that is something that can be very challenging. So rejection is a serious concern. Obviously, people take immunosuppressors and things like that. And yes, that doesn't even have anything to do with any complications associated with the surgery or anything like that itself. So yeah, so again, there's many factors that go into it. So I'm not sure if there's a meaningful percentage or something like that of how many successful or how successful things like that are. Yep. All righty. One last important question, and this does kind of piggyback a little bit on the transplants. When it comes to healing, how regenerative is cardiac muscle?
It's not very.
Yeah. Yeah, basically there's no regeneration of cardiac muscle. When we damage our cardiac muscle, that cardiac muscle pretty much stays damaged, right? And if the damage is severe enough where it disrupts the normal neural activity, that affects the function of the heart. And you have to get something like a pacemaker or something like a transplant, otherwise you won't survive. All right. We've
So-
done it all here with all the pretty words. We can look at the lecture and do the same thing with all the pretty words. So again, we've got our skeletal muscle, which we know is connected mostly to bones. Is striated, is voluntary, is multinucleated. Why cardiac muscle doesn't? Again, why is always one of those questions that's hard to answer in anatomy and physiology, because again, the engineer who designed all this did not leave us her footnotes. What I would say is that research appears to show that it's primarily due to the activity. As we learned, mitosis, dividing new cells to make new cells, requires a tremendous amount of resources and a tremendous amount of energy, right? The problem is that our heart rate is constantly active. And because it is so active metabolically, it's probably very challenged for it to, take a Sunday off where it has time to divide. So my guess is that it's probably due to the activity of it. Sydney, I think you were first. Let's go ahead and.
So it's not that it just doesn't regenerate at all. It's not that it repairs itself by fibrosis, but it doesn't.
No, so okay.
Or would it be fibrosis?
Yeah, it would repair itself by fibrosis. And so here's what I'll say. Remember, in this class, the sky is blue, right? In this class, cardiac muscle does not regenerate. However, I will tell you that in the past 15 years, there were two really interesting studies that came out. One of them involved Chernobyl, right? For those of you who remember, back in ancient times, Chernobyl had a nuclear plant that lost its failsafe and erupted, and many people received massive doses of radiation as a result of that. And in looking at the long-term implications of those, one of the things they've been looking at is the deterioration of by looking at the radiation rates and the half-lives of the radiation in the people's bodies, right? They can follow, because again, radiation deteriorates at a very specific rate, that half-life rate. And what they found is actually in most of the people that they looked at 10, 20, 30 years later, they found that there was less radiation in the heart than they expected, which was an indication that there may have been some small amount of regeneration of that tissue taking place. The other thing goes back to kind of what was mentioned before. If with heart transplant, getting the right heart is something that can be very, very challenging for a transplant standpoint. So one of the things that they will either sometimes do temporarily is they will do a piggyback surgery, where what happens is if they can't get quite the right heart, but they can find something that's functional, they will not actually remove your heart, but put a second heart into your chest to help to assist in the pumping of blood. When they went back in and removed that second heart, one of the things they found in a couple of anecdotal cases is that some repair had occurred in the original heart. So it seemed like taking that some of the workload off of that first heart allowed it a chance to regenerate, right? These long-term 30, 40-year types of process show there may be some slow regeneration that can occur. But again, these were just two very simple studies that occurred, have not been recreated or anything. So not surprising, like most things we've talked about in this class, everything is more complicated than we've talked about. And so there may be some very limited, very slow, or in certain conditions, ability for cardiac muscle to repair itself. But for all intents and purposes, and more importantly for the exam in this class, cardiac muscle does not regenerate.
Got it. Thank you.
Wow, that's kind of terrifying, Julia.
I think I made a mistake. He must have died a lot later than 50 because my dad is 50.
Yeah,
So
okay,
probably 70, but like they kept expecting him to develop something scary suddenly, but it was a very slow, long process.
Well, still very tragic, so I'm sorry about that. All right, so we did all of that, we did all of that, we did all of that. So again, controlled by hormones in the autonomic nervous system, influenced or modified by hormones of the autonomic and hormones in the autonomic nervous system. And of course, skeletal muscle is controlled by the somatic motor nervous system. We did all of that. I also promised you some histology. So notice here, we can look at using a light microscopy. We can see, and again, depending on the size of the screen, hopefully you can see this. So what I will do is, let's use black. Notice what we're seeing here is a portion of 4 cells. So here is some skeletal muscle, 1, 2, 3, 4 cells. Notice for the 4 cells, we can see the division between the 2 cells. And notice we can see how the nucleus in this is pushed to the side. This one's nucleus is pushed to the side, right? This one's nucleus is pushed to the side. And again, we're seeing just a small portion. This is not the ends of it. So that's why we're only seeing a few nuclei on this. But the other thing that I want you to see very clearly here is the striations. Notice there is a region here that is lighter in color, and then an area that is darker in color, and then lighter in color and darker in color. This is because of the precise arrangement of those proteins. And notice, if you look closely at this picture, the other thing that you'll see is in the middle of the light region, there is a slight darker band. In the middle of the lighter region, there's a slightly darker region inside that lighter region. And if you look really closely at the center of our dark stripe, there is a little lighter region in the center of the dark stripe. You guys able to see that? Hopefully you have the magnification. This is because of the precise arrangement of proteins. And what's really, really awesome, if you look at it, is you notice the light stripe from one lines up with the light stripe of the next. These proteins are so perfectly arranged, not only are they perfectly arranged within one muscle cell, but they're perfectly arranged across all of the muscle cells. Why might this be? Yeah, these are the dark band and the light band, absolutely. You're not going to see mitochondria with a naked eye, well, not a naked eye, but with a light microscopy like this. To see mitochondria, to see that type of organelle, you need an electron microscope. So you're not going to see it at this magnification. Okay. Yeah, so again, the dark regions and light regions are the I bands and the A bands. And then we'll also talk about the H zone and the M line and the Z line and all those types of things as well. Well, if you tear a muscle, then hopefully you give it a chance to repair. Although if the damage is severe enough, then it could cause some fibrosis to occur, and that could affect the function of the muscle. So yeah, so it can be a concern that way. All right. Yes, so yes, what this muscle cell, as we'll see, is filled with is these organelles that run the entire length of the muscle cell. And obviously these are not straight lines, but they're so precisely aligned that they form the proteins. And trust me, we're going to take this and break it down to see these individual proteins. So absolutely, we are going to go far, far deeper into this muscle cell and see how this works. Not this type of a microscope, probably, but an electron microscope, you probably could. Yes, technically, yes, but soreness can be caused by lots of other things that you wouldn't necessarily see from the proteins either. But I promise you, we're going to break this down. So we've looked at this and we'll look at it in much, much further detail. But again, if you think of our characteristics that we've talked about for skeletal muscle, what shape did we say skeletal muscle cells were?
Cylindrical.
There you go. Excellent. Where is it found?
On your bones.
Attached to bones.
Mostly attached to bones. Absolutely. Voluntary, yes or no?
Yes.
Yes. Striated, yes or no?
Yes.
Excellent. Number of nuclei per cell, one or many?
Many.
Many. And able to regenerate?
Yes.
Yes, but limited.
Limited.
Right? Excellent. Compare this to smooth muscle. Now notice, as I mentioned, these spindles interdigitate with each other to form a uniform layer. So one of the things that we can do, and one of the things that you will be responsible for, is a teased prep. With a teased prep, it's just like it sounds like. They use a couple of needles and they'll come into the tissue like this, and they'll pull the cells apart from each other. And when they do that, you can actually see, so you can't really see it, but we can pretend to see it here, that we've got this spindle-shaped cell that comes out and tapers and forms something like that. So again, as we talked about, this has that spindle shape with that centrally located nucleus. Where is smooth muscle found?
Lines, hollow organs.
Yeah, in the walls of hollow organs, mostly. Again, there are some exceptions to that. Voluntary?
No.
No.
Striated?
No.
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 Number of nuclei per cell, one or many?
One.
One.
Excellent. Able to regenerate?
Yes.
Yes.
Yeah, this one has the highest rate of regeneration. Excellent. And lastly, cardiac muscle. Shape.
Branched.
Branch. Right. And again, notice here we see a great example of one of these cells. Notice this one has 1, 2, possibly a third branch to it on this side. So here we see an example of a single cardiac muscle cell. Notice I can easily tell where our cardiac muscle cells end because there is this dark line. This dark line is what is known as the intercalated disc. And it is the junction where one cardiac muscle cell connects to another cardiac muscle cell. Now, if you remember when we talked about cell junctions way back in the day, this intercalated disc contains massive numbers of desmosomes and gap junctions. Someone remind me again what desmosomes do?
You know, I forgot about that one.
Are those the ones with the little spindles at the end that send signals from one side to the other?
So close. They do have those the spindles, and they can actually have fibers that go from one cell to another. But remember, they form that really strong spot welding that holds the cells together really, really strongly. Right? Your heart has to generate a massive amount of force to pump that blood through your body. And so being able to lock these cells together is very, very important. What did gap junctions do?
They help
Act.
communicate.
Yeah, they formed a fluid-filled tunnel between the cells. So whatever happens in this cell, so for instance, if this cell forms an action potential, that action potential can spread to all of its neighbor cells, allowing these to work together as one. Exactly, I like that. It electrically connects the cells so that that electrical signal, that action potential, can spread through all of it so that all the muscle cells work together as one to form a very efficient pump. All right, excellent. Now, great question. Notice with the other advantage of this branching shape is there is a lot of space in between. Notice, in fact, the space in between has nuclei in it. This stain doesn't show the nuclei of the cardiac muscle cells. All these nuclei that you're seeing are all the nuclei of the connective tissue in between. This cardiac muscle actually has what we call a fibrous skeleton. Oops. A large amount of connective tissue that helps to hold and give structure to the shape of the heart. So within this muscle layer, there's a lot of fibrous connective tissue, and that's what that white stuff is. Many years ago, remember we were just talking about how hard it is to get a heart transplant. So one of the things that they started trying to do, because they thought it was a really interesting field of study, is they would take hearts out of cadavers and they would dissolve away all the muscle tissue. so that all that was left was this fibrous connective tissue, this fibrous skeleton. And then what they would try to do is use a person's cells, their own stem cells, to try to create cardiac muscle on this model so that you could basically grow a new heart on this model of a heart that could then be used to put in the body. And because you use the own tissues from that person's body, it would be much less likely to be rejected. Now, what's even more interesting is they don't do this research anymore because it isn't something that's viable. Why aren't they using the fibrous skeletons for this anymore? Why aren't they ripping the hearts out of cadavers and doing this anymore? Anyone know? No one's heard of this?
Probably like human rights activists.
No, nobody cares about humans. We all care about puppies and other cute things like that, but nobody cares about humans. So that's not it.
Did it not work?
It's challenging and hard, but it's something that there is showing promise in. However, they decided that getting these fibrous skeletons wasn't as important anymore because God created 3D printers. And so what they would do instead is they would 3D print a skeletal model and then try to use that to populate with the cells instead. So the same research is still going on, but instead of using the fibrous skeletons from cadavers, hearts, they're actually printing 3D models of the heart to try to populate those and grow a heart that way.
That's pretty cool.
It is. Yep. Science every once in a while is very cool. Yulia, did you have another question or is that hand up still from before?
It's from before.
I'll go ahead and lower that just because so that I know when you guys are asking. All right, excellent. So here we've got, as we mentioned, that branched shape. Of course, where is cardiac muscle found?
Only in the heart.
Heart.
Only in the heart. Excellent. Voluntary, yes or no?
No.
No.
Striated, yes or no?
Yes.
Yes, mostly.
Yeah, no, not mostly, definitely. It is striated. We can definitely see the striations. And notice the striations run parallel to those intercalated discs. So intercalated discs and the striations, the stripes, are oriented the same direction, but you can clearly see that the intercalated discs are much darker, much larger than the striations. But we absolutely, positively have striations on this that are separate from the intercalated discs.
I have a quick question. Is it important for us to know about these intercalated discs and what exactly that means? Are both of them, so is inside them where we have the gap junctions and the desmosomes?
Yes.
Okay, how important is it for us to know this?
Well, seeing as you have a study guide that has histology on it, and when you look at that study guide with histology, it definitely shows cardiac muscle histologically. And one of the things you're responsible for is intercalated discs. And knowing what an intercalated disc is definitely something that you would need to understand because it involves cell junctions that we talked about in the earlier part of the class. I would say with 100% certainty that it absolutely is something that you need to know.
Okay, thank you. And they contain many different components to them, right? Not just the gap.
No, the intercalated discs, yes, yes, there are other components to it. However, what it contains the most of is those desmosomes and those gap junctions.
Okay, thank you.
Yep. Excellent. Number of nuclei per cell, one or many?
One.
One. And again, you guys are right. There are some cardiac muscle cells that may sometimes have a second, but for most heart muscle cells contain one. Most contain one. Occasionally you'll have two, but definitely not as many as we see in our skeletal muscle. And then this one able to regenerate?
No.
Exactly. We're going to stick with the company line on that. Excellent. So to answer the original question, absolutely. And in fact, if we cheat and go there, look in our modules, look at our muscle histology, Again, notice we need to be able to recognize that T's smooth muscle and some characteristics, and then a cross section through an intact organ. I use the stomach, but any hollow organ that has smooth muscle and it would be able to show you these layers. Cardiac muscles, where we need to be able to recognize the intercalated discs, right, and things along those lines. We need to know all of that, and our skeletal muscle and a striated and cross-sectional view. Neuromuscular junction we'll talk about later. So absolutely there is some histology here that you absolutely positively are responsible for. So make sure you are comfortable and familiar with this material. That is some, but not all of what we need to know. All right. So absolutely positively for the lab exam, for the lecture exam, you need to know this information. However, from after the break moving forward, we are primarily going to focus on skeletal muscle. The majority of the rest of this section, we are going to focus on everything you ever wanted to know and more about skeletal muscle tissue and the skeletal muscle cells of our body, right? Eventually, on the last day of this section, we will come back, and after we've learned everything you possibly there is to know about skeletal muscle tissue, we'll come back and compare it to smooth and cardiac to get some ideas of the similarities there. But from now forward, moving forward, our focus, not in the same kind of way. There's going to be something similar. You're going to have to do something similar to it, but not in the same type of way. No. So yes and no. We're going to focus on everything you ever wanted to know about skeletal muscle and more. So let's go ahead and take our first break. What time is it now? 1:22. So let's come back at 1:37. And at that point, I will start the recording. And we are going to dive deep into the anatomy. We're going to take a muscle like your bicep brachia and break it down bit by bit by bit by bit till we get to the point where we are looking at individual proteins. All right, so that is going to be a tremendous change in scope from going from bicep brachia down to individual proteins inside of a single muscle cell. That also means there's going to be a tremendous amount of vocabulary, right? One of the advantages of that pre-lab, and again, hopefully you're not just blindly doing the pre-lab, but actually looking at the material and starting to make some sense of it. But we will talk about, we're going to get into, there's a lot of anatomy on this that we're going to get into. The pre-labs may not have a lot of information in it, but you have your textbook and other resources, your modified mastering A&P, the almighty Google, plenty of other things for you to be able to find. And that's the point. I wanted you spending the past 5 days thinking about this because it's going to be hard. There is just as much anatomy in this section as there was in the skeletal section, but there's even more physiology. So this section isn't as hard as I think the tissues one is, because that one's less intuitive, but it has a lot of material. This section probably has the most material in it. And if you look at it, I think it's the one that we have the most lectures for, or actually this in the nervous system. So those two, we have similar number of lectures, similar amount of information. So excellent. Yeah, and that's what you should be doing for those pre-labs, looking at this material and understanding the material. All right, excellent. Let's go ahead and take our break then, come back in. Actually, we've lost 2 minutes now. We'll come back. I want to get your full break. So, because again, we need to be fresh because we've got a lot of vocabulary to go with. So let's come back at 1:39, and at 1:39, we will start from there. All right, see you guys in 15 minutes. So, as I mentioned, any questions before we get started? Because like I said, from here, we start getting deep, literally. All right, excellent. So when we talk about a skeletal muscle, and in this case, I'm talking about the organ. And someone remind me again, how many named skeletal muscles did we say we have?
600.
Yeah, over 600 of them, absolutely. So when we talk about one, like the bicep brachia, for instance, that skeletal muscle is made up of somewhere between hundreds and thousands of muscle cells. Now, as we've talked about, muscle cells are long cylinder-shaped cells that can be as much as a foot long. And so the term muscle cell and the term muscle fiber, because it's basically like a nice long thin fiber, are pretty much used interchangeably, and I will use the terms interchangeably as well. So muscle cell, muscle fiber mean the exact same thing. Now, that doesn't mean that you can take thousands of these muscle cells and dump them into a bucket and you suddenly have a bicep brachia. No, they need to be precisely arranged. And to do that involves many layers of connective tissue. And so these connective tissues help to support them, hold them together, provide protection, provide support, and all of that type of stuff as well. Now, also, right, when you get muscles, individual muscles are organized as well. If you've got 600 muscles in your body, you need to organize this together, right? And many of you have experienced this in certain ways, right? Again, if we have time at the end of this section and you want to get into the deep philosophical debate as to whether or not meat is murder, we can definitely have that deep philosophical discussion for those of you who are vegan or vegetarian. However, the one thing that you cannot debate is that meat is muscle. So when we talk about this, for instance, if you, back in ancient times when we were allowed to go outside, went to the Renaissance fair, of course, the primary reason you go to the Renaissance fair is to get that big, huge turkey leg that you can eat. And when you take a bite off that turkey leg, does the meat just peel off as like a big, huge layer of foam that comes out that way?
No.
No, right? It comes apart in individual pieces, right? And the reason for that is that there are connective tissues, fascias, that help to define and distinguish the muscle, right? Separating muscles from each other, supporting muscles and allowing them to do their job. One example of this is a structure we've already talked about. Remember, we used terms like hypodermis. We use terms like subcutaneous layer to talk about that layer of areolar connective tissue and adipose that sits between our skin and our muscle. Remember, we said it was not a part of the skin, but under the skin. Well, we can give a third name to that component. That hypodermis, that subcutaneous layer, is also known as the superficial fascia. And as we talked about, it allows the skin to loosely connect to the muscles so that as you contract the muscles and they move around underneath the skin, it doesn't pull on the skin and put too much stress on the skin. But anybody who's ever eaten a turkey leg or deboned a chicken knows that individual muscle groups are going to be separatable from each other as well. So there are deep fascias. For instance, there are pockets in my arm where the muscles that are responsible for flexing my elbow and the muscles that are responsible for extending my elbow have their own connective tissues that wrap around them so that they can move separately from each other and don't interfere with each other with their function. And so again, if you've ever deboned a chicken, it doesn't just peel off the bone. You can actually pull off individual muscles and pull it off that way. I wouldn't say necessarily the more meat you eat, the more muscle you get. Again, it's really dependent on using that muscle. The more you use the muscle, the more you'll build. But absolutely, you need proteins. Meat is one of the primary ways to get proteins, but there are other ways to get proteins as well. But obviously, meat is a big one. All right. So this is how muscles work together. Well, okay, I wouldn't say, it depends on what you're using that protein shake for, right? If the goal is to, anytime you ingest a massive amount of anything, right, there's obviously going to be limits on how much of it can be absorbed into your from your digestive system into your body. So that's what I would say from that. So, but again, again, I would say that it varies depending on how you're using them and what you're using them for. All right. So let's talk now about an individual muscle. Again, any of those 600 muscles like the bicep brachia, although clearly this isn't the bicep brachia, because what muscle is this connected to? I mean, what bone is this connected to?
Femur.
Femur, absolutely. So obviously this is a leg muscle of some sort. We can see the arrangement of the muscle cells. Here we see a single muscle cell pulled out here, and we can see how they're arranged to make a skeletal muscle. So let's do this. Let's actually bring back up the whiteboard. And talk about how you would make a muscle. So the first goal, like we talked about, is to have a muscle cell. And actually, let's go back. Let's do this. Cheat and do it this way. So here's a muscle cell, and a second, and a third. Each individual muscle cell is going to be surrounded and protected by a connective tissue. This connective tissue that is going to wrap around and insulate and isolate each individual muscle cell. Right? Remember, one of the things that we said are these things are electrically excitable. They produce electrical signals. They carry electrical signals, kind of like the wires in your wall. And if you opened up your walls, would you just see a whole bunch of bundled copper just hanging out together? No, you would need to insulate them and isolate them from each other. And so that's what we have here. We have this connective tissue that happens to be areolar connective tissue that wraps around each individual muscle cell. Now, obviously, this is not the only areolar connective tissue we find in the body. So, based on location, we can give a name for this one, and this one is the one that wraps around individual muscle cells, and it is known as the endomysium. So based on location, we call this areolar connective tissue that wraps around each individual muscle cell. We call it an endomysium because it is within the muscle. Now, these individual muscle cells, this muscle cell and 10 of its closest friends are going to be bundled together into a structure. And that bundled together structure is what we call a fascicle. So here we've bundled them together to form a structure that we call a fascicle. And this fascicle is a bundle of muscle cells. Now, obviously, to make this bundle of muscle cells, we need a connective tissue to wrap around them. And so we use our dense irregular connective tissue. Obviously, this is not the only place we find dense irregular connective tissue. So again, based on its location, we call it a perimysium. And this fascicle and a bunch more fascicles are going to be bundled together as well. And again, you may not have ever thought of a fascicle in this term or how these muscle cells are arranged in any fashion this way, but I guarantee most of you are absolutely positively aware of this. Because, right, with the weekend approaching, one of the things you may be doing right now is marinating that wonderful tri-tip. All right, putting it in that dry rub on it, marinating it for 3 days in the bottom of your fridge, bringing it out to temperature, getting it on that nice hot grill, searing it, bringing it to temperature. You put all this time, all this effort, all this love into it. You let it rest the 20 minutes to reabsorb the juices. And after all that work and effort, do you just close your eyes and randomly cut it however the hell you're going to cut it?
Nope.
No.
If you get that really nice tri-tip that you spent a lot of money on, how do you cut that tri-tip?
Against the grain.
Against the grain. Well, guess what the grains are? The grains are these fascicles, right? Connective tissue. Think about it. When we cut this tri-tip, we want to cut across the grain because it breaks up the connective tissue. Right? If you cut it longitudinally with the grain, you're getting all that connective tissue and it's not going to be nearly as tender. But maybe you don't have the money for an expensive tri-tip. So instead you buy that cheap chuck roast and you put it in the crock pot and you pour a can of beer over the top of it and you let it sit in there for 12 hours. What happens to that meat after all that time?
It tenderizes.
Okay, and so when you go to pull it out, does it come out as just one big solid mess? No, it shreds, absolutely, right? But when it shreds, does it form these irregular shaped pieces?
Kind of.
Yes and no. Yeah.
Well, yes and no, but when you pull it out, it shreds into these thread-like structures, right?
Yeah.
Again, those thread-like structures are these fascicles. Basically, when you cook this low and slow, the collagen in this dense irregular connective tissue breaks down. And as it breaks down, the individual fascicles shred and come apart. So again, you may not have thought of fascicles before, but we're all aware of, the grain of a meat. And when we talk about that grain of the meat, we're talking about the arrangement of its fascicles. Right? Made by that dense irregular connective tissue that forms the perimysium. I'm not sure what you mean by black lines. Ariana? Well, maybe she's typing. Laura, while she's doing that, I'll go ahead. What's your question?
Yeah, so you said the structure or the fascicles, what was the word that was also, it sounded like the word fascicle? You said the black lines were,
So the black lines, so I see. Oh, those black lines. No, so okay, I understand now. No, these black lines here are individual muscle cells.
okay.
So inside, so let's actually do that. Inside of every single one of these fascicles is somewhere between 10 and dozens of muscle cells. So every fascicle is comprised of parallel muscle cells. So all of these contain muscle cells inside of them, and all of these muscle cells, and again, I'm going to cheat and do this way. All of these muscle cells have that endomysium, that areolar connective tissue that wraps around them. So we have areolar connective tissue separating and isolating each individual muscle cell. And then this muscle cell and dozens of his buddies are all bundled together by a dense irregular connective tissue to form a fascicle.
Oh, okay, so the long black lines, they're muscle cells. They're
Those
not
are the muscle cells. Yes, those are the muscle cells inside of the fascicles. Now, of course, we need to bundle these fascicles together, but we're also going to bundle them with blood vessels and nerves. So in between the fascicles, we'll have nerves and we can have blood vessels. So we'll have and all of these structures, the fascicles, the blood vessels, the nerves, all of those are going to be bundled together. Oops, nope, don't want red. For you. Are going to be bundled together by yet another dense irregular connective tissue. And they're bundled together to form the muscle. And of course, this dense irregular connective tissue that forms the muscle that wraps around all the fascicles and wraps around the blood vessels and wraps around the nerves, based on its location, we call it the epimysium. No, muscle cells do not get replaced the same way that bone cells do. And remember, it's really not the bone cells that are getting replaced, it's the bone matrix that gets replaced. All right. Again, my drawing is very, very simplistic, but let's go back and look at the picture here and we can see all of these same things. And let's work our way down. Notice in this case, we have an entire muscle made up of many fascicles and also some blood vessels and nerves in here as well. So this connective tissue that is wrapping around the entire structure is that dense irregular connective tissue that we call the epimysium, filled with all those fascicles. Notice they've pulled out one of the fascicles here. This fascicle is, of course, formed by many muscle cells, all bundled together by another dense irregular connective tissue that we call the perimysium. And this structure that is made of all these muscle cells bundled together, remember, we call a fascicle. And notice inside of this fascicle, we have many muscle cells, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, so on and so forth, individual muscle cells. But notice if you look closely, and let's use bright green for this, see if we can show this off. Notice in between the muscle cells, we can see a little bit of connective tissue separating and isolating each individual muscle cell. This connective tissue between each individual muscle cell is a relar connective tissue, and we call it the endomysium. So notice we see that with this picture. This illustration shows it as well. Notice here we have a cross section through an entire muscle, the organ like the bicep brachia. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 fascicles, all bundled together along with blood vessels and nerves. That is what a muscle looks like. And it is formed by that dense irregular connective tissue that bundles it all up, known as an epimysium. Of course, notice we can grab one of those fascicles and pull it out. Notice this particular fascicle is comprised of 1, 2, 3, 4, 5, 6, 7, 8 individual muscle cells. These 8 muscle cells are bundled together by a dense irregular connective tissue that, based on its location, we call a perimysium. Notice also we can see that each individual muscle cell is surrounded by an areolar connective tissue that we call the endomysium. Now notice there's still a whole lot more going on inside the muscle cell, so we have to go even deeper. But here we are breaking down how you make an organ that is a muscle and the connective tissues that arrange all of these. All right. And again, we can see this on illustrations, we can see this on models, but we can see this with the microscope as well. Notice with this light microscopy, we can see the epimysium that is wrapping around everything. Notice here, we can see an individual fascicle, a bundle of muscle cells that are bundled together. And remember, both the epimysium and the perimysium are dense irregular connective tissues. One forms the muscle and it's on the outside, so we call it the epimysium. One's on the inside and forms the fascicle, so we call it the perimysium. Notice the muscle cells are red, but in between the muscle cells, we can see this white connective tissue that is separating each individual muscle cell. And that is our endomysium, that areolar connective tissue. And there's one more cool thing about this as well. Let's go back to this. We have all these tissues, these connective tissues, and these connective tissues run the entire length of the muscle cells. But not only do they run the entire length of the muscle cells, but they actually project beyond the muscle cells. So if you think about it, the collagen fibers from the epimysium extend beyond the length of the muscle cells. The collagen fibers from the perimysium, from the fascicles, extends beyond the length of the muscle cells. And even the collagen fibers of the areolar connective tissue that forms the endomysium extend beyond the length of the muscle cells. Notice as all these collagen fibers extend beyond the muscle cells, and let's make this a little smaller so it fits. Beyond the muscle cells, notice all the collagen fibers start to run parallel to each other. And when they start to run parallel to each other, what structure are they forming out here?
Maybe ligaments or tendons?
There you go. Absolutely. Because they're running parallel to each other, they form a dense regular connective tissue. And you're absolutely correct. This dense regular connective tissue is going to connect this muscle to the bone. It forms the tendon. And that's what we see here. Notice one more illustration. Single muscle cell with an areolar connective tissue wrapped around it that we call that endomysium. This and 12 of his closest friends are bundled together into a fascicle by a dense irregular connective tissue we call the perimysium. This fascicle and 6 of his closest buddies, along with blood vessels and nerves, are wrapped up by a dense irregular connective tissue called an epimysium. Yeah, I'll go back to it in a second. And the collagen fibers from all of those extend out beyond the muscle cell to form the tendon that is going to attach it to the bone. There you go. And I'm not sure how much point there is in it, but I can always save this and add it to the website if you like. All right, to get what you needed from this. Again, I fully appreciate what an amazing artist I am, but honestly, I think looking at something like this makes more sense to me, but I'll happily post that if you guys want it. All right. Questions on how we arrange our skeletal muscle cells to make a muscle. Because again, if it's not making sense now, it's just going to get a whole lot worse. So let's make sure we understand this. So questions on this? Well, I did say they were amazing, so there. All right. Any other questions on this?
So like when you say based on location, is it based on where it's at in the body, or is it always going to be the perimysium, the epimysium, and the endomysium?
But that's the point. That is exactly where it is in the body. Think of it this way. If I had a tiny little scalpel and I cut off a little bit of this dense irregular connective tissue and a little bit of this dense irregular connective tissue, and just those dense irregular connective tissues I looked at under a microscope, they would be identical.
Okay.
Because they're dense irregular connective tissues. So why do we call one an epimysium and one a perimysium?
Oh, gotcha. It's because one surrounds the whole muscle fibers and the other ones just surround a group of them.
Yeah, exactly. Their location. This dense irregular connective tissue is bundling up muscles to form a fascicle. This dense irregular connective tissue is bundling up fascicles to make a muscle. So the tissues are identical. The only difference is their location and therefore their job. And so that's why we give them special names. And that's why I use that term based on location, because that helps us to be able to differentiate what it is that we're looking at. So on an exam, again, I could have this exact picture with an arrow pointing right here, and I could basically ask you two questions. Identify the tissue type, to which of course you would say,
Dense irregular connective tissue.
or I could ask you to identify the tissue based on its location, in which case you would say,
epimysium.
there you go. See, just that easy. Yulia, you had a question?
Yeah, so does our body build more muscle cells, like if we're working out, or do the existing ones get bigger? Or like, I felt like I never got a full answer, like, do we ever replenish our muscles or no?
So we are going to get to that. I promise you we're going to get to that. The short answer is, for most growth, we are not adding or subtracting muscle cells. We are increasing or decreasing the size of the muscle cells. And we'll see exactly why in just a minute. We're literally going there next. We're literally going there next. Okay? So hopefully that will help to make some sense of it.
Okay, thank you.
Excellent. All right. So, of course, tendons, we know, connect muscle to bone. And remember, we've also talked about that term, a big, broad, flat tendon. So, for instance, in your abdominal region, you have a big, broad, flat tendon over the top of your head. You have a big, broad, flat tendon. And because anatomists love to name pretty much everything, we have a name for a big, broad, flat tendon, and that is an aponeurosis. Aponeuroses would be the plural, meaning you have more than one. Aponeurosis is the singular. So again, just fun with vocabulary. All right. So as we said, we have been breaking down. Here is our skeletal muscle. That is the organ, right? Comprised of fascicles. So we pull out a fascicle and we looked at that. That fascicle is comprised of muscle fibers, muscle cells. So we've looked at that. But as you can see from the illustration, there's a whole lot of stuff going on inside of this muscle cell as well. So now we're lowering our view, focusing our view, going in even deeper, and we're going to look at everything, the organelles and even the proteins and how they're arranged inside of our muscle cell. Now, before we can fully do that, we do have to talk a little bit about how we make muscle cells. And this will kind of answer the question you were asking before, Yulia, and a couple of others of you were asking as well. How we make muscle cells. Remember, we had pluripotent stem cells. Like the mesenchymal cell, that are capable of producing all of the cells associated with connective tissues. Well, muscle is not connective tissue. So not surprisingly, it is a completely different stem cell, a stem cell known as a mesodermal cell, that is a pluripotent stem cell. Oops. That is used to make muscle cells, skeletal muscle cells. And the immature cells that we use to make muscle cells are myoblasts. Now remember, one of the things we've talked about with our skeletal muscle cells is they contain many, many nuclei. And that is because hundreds of these tiny little myoblasts fuse together to form a single cell. So what we call a single skeletal muscle cell or a single skeletal muscle fiber is a very complicated structure. That is made up of hundreds of cells fuse together. It's one of the reasons why regeneration is so limited. If you have normally we know we make more cells by dividing a cell. One cell becomes 2 cells, and then suddenly our tissue has grown. But if I have a single cell that is made up of hundreds of cells, that's not something that can just use mitosis to split itself in half. And suddenly we have 2 skeletal muscle cells. It's far more complicated than that. And that's also true, not just as we continue to grow and age, but as Julia pointed out, right? It's exhausting. We're down, we've lost a third of the students. I mean, nobody's dropped yet, but a third of the students in this class aren't here right now. They're tired, you're tired, I'm tired. We've all been stuck at home for forever. So there's a very strong desire for us to just chuck it all. And instead of continuing to do anatomy and physiology, we may decide that we want to just work out for 4.5 hours every day. So when we meet 4.5 hours every day, we're going to work out together 4.5 hours today, motivate each other. And for the next 10 weeks, that's what we're going to do. Now, at the end of those 10 weeks, are we all going to have bigger and stronger muscles as a result of that?
No.
No? You don't think?
Well, some are. I feel like based off like your biology, some people aren't able to.
You are correct in that everybody will not gain the same amount of muscle mass, but I'm fairly certain that if we all worked out 9 hours a week for 10 straight weeks, everybody would at least get stronger and their muscles would get bigger. Yes, some will have bigger muscles than others, but everybody's muscles will get bigger. However, not because we made more muscle cells. Instead, what actually happens is our muscle cells make more proteins. And when our muscle cells make more proteins, the muscle cells get bigger. So what happens is our muscle cell makes more proteins. And when it makes more proteins, the cell gets bigger. So if we're all working out 9 hours a week for 10 weeks, our muscles are going to get bigger because the muscle cells got bigger. This type of growth is what we call hypertrophy. Right? And while some of you may not have necessarily worked out for 9 hours a week for 10 straight weeks, maybe some of you have broken a bone in your arm and had to wear a cast for 9 weeks. And when that cast came off your arm after 9 weeks and you compared your 2 forearms to each other, how did they look?
The one that was in the cast had smaller muscle.
Absolutely. Notice without use, what ends up happening is muscle cells get smaller. Yeah, one could be more hairy as well, absolutely, or at least the hair is darker because it hasn't been exposed to the sun, right? And in that case, the muscle cell gets smaller. So with lack of use, we break down those proteins. Remember, as we said, our muscle is a nutrient reserve. If you're not going to use those muscles, we're not going to let you keep all those proteins inside of there. And so what's going to happen is we're going to start breaking down those proteins and the cell gets smaller. And of course, what do we call that process?
Hypertrophy?
That's a good guess, although that's not the term. When they say, oh, your arm got smaller, it
Atrophy.
Atrophied. Atrophied. Absolutely. So typical change in size of muscles is due to the change in the amount of proteins inside of it, allowing them to get smaller or bigger. All right, so far so good? Now, I do want you to notice one more thing about this illustration. Notice we have these myoblasts dividing, fusing together to make our muscle cell. And notice most of those myoblasts fused and formed a skeletal muscle cell. But what's the key word in that sentence? Most, because most means not all. With a muscle cell, typically, a few of those myoblasts stay immature and just stay next to the skeletal muscle cell. And I do mean next to. They are actually inside the endomysium. So right there, right next to the muscle cell. These cells become what we call satellite cells. And these satellite cells are the ones that allow us the limited regeneration. If the muscle cell is damaged, these satellite cells can divide to make new myoblasts, to repair the damaged cell. So notice if one muscle cell gets damaged, I can divide myoblasts to either repair or even replace it if necessary. However, if that shark takes a big, huge chunk out of my arm, am I going to have enough myoblasts to be able to replace all of those muscle cells that were lost? No, of course not. And so as a result of that, then it will heal via fibrosis. There, I'll get the scarred tissue instead. So notice we can heal one cell with its myoblast, but we can't replace a whole arm. So originally the myoblasts were the immature cells that fused together to form the muscle cell. And the remaining myoblasts that we call satellite cells are the ones that are going to repair the muscle cell.
So do we actually have a picture of a mature muscle fiber on the screen or no?
It's working its way to mature, but you can tell that this is what is going to become the cylinder shape skeletal muscle cell, and this is the satellite cell that's going to be sitting next to it.
Okay,
If
thank
you
you.
actually want to see a nice illustration of this, notice when we come over here to our modules and we look over here, here we have muscle classroom models. This nice handout that I've put together. We have individual muscle tissues. What type of muscle cell do you think that is based on the information we have?
Skeletal.
Skeletal
Yep.
muscle. Excellent. What's that?
Smooth.
Smooth. What's that?
Yeah.
Cardiac. Excellent. But notice what we have here. Well, let's see a different view. Hold on. Why are you not? Oh, there we go. This one's the one I wanted. It's not fully loaded yet. Can you guys see this?
Yeah.
Yeah.
A little fuzzy, but it'll load eventually better. But what we have here is a cross section through a single muscle cell. If you look closely, you'll see this is the plasma membrane of it, wrapping all the way around and coming around to the back and coming in this way. Here we see the nucleus pushed off to the side, the organelles and stuff that we're going to talk about in a minute. But notice also, if you look closely down here at the bottom, you can see the endomysium, the connective tissue that is wrapping around it. And right here, between the endomysium and the cell itself, is this blobby little cell right here. Guess what this blobby little cell right here is?
Satellite cell?
That's the satellite cell.
What
Excellent.
about to the right? What's the yellow structure?
I don't know. I've never seen that before in my life. I'm just kidding. It's the neuromuscular junction. It's a specialized area for communication that we'll get to there.
I thought he was serious for a second.
No, I was not. Here, oh, here we go. This is the view I wanted. Notice here we see it straight on. So notice again, here is that endomysium wrapping around it. Here we see the plasma membrane of our muscle cell and all of its specializations, which we're about to talk about. And then right here in between is that satellite cell. So we actually see that satellite cell right here that is going to allow this to repair. Well, I know everything about this because I've taught it for 20 years, something crazy like that, that I've been teaching anatomy and physiology. So I've had more practice than you have. So that's the only reason why I know everything. You know, if you ask me something about plants, I don't know jack about plants. Plants are stupid. So, but yeah, this I know a lot about, but just because I've practiced more than you have. Trust me, if you'd taken this class 20 times, you would know just as much as I do. All right. So here's a nice model that shows that. And not surprisingly, this is a model that's in the classroom and definitely something that is going to be on the exams. We have another model. Hold on, I got to clear the picture drawing of a single skeletal muscle cell. And again, here we see the plasma membrane. Here we see, I think I've got a side view from it. Maybe not. It's got this gray matter over here is the endomysium that wraps around it. But notice this one doesn't have a satellite cell. But we'll get to, what we have to do next is talk about all the other things. Now, there's one other thing that I wanted to show you as well. Hold on a second. Let's switch to this view. All right, hold on. All righty. So again, these satellite cells are going to allow for that limited regeneration that takes place. And like we said, we all work out for our 9 hours a week. For our 10 weeks, our muscles are going to get bigger because they grow more proteins. They get bigger via hypertrophy, not by making new cells. If we were to make new cells, that process would be what we call hyperplasia. Now, if instead of working 9 hours a week for the next 10 weeks, we decided to work out 9 hours a day for the next 10 weeks, then not only would your muscles get bigger, but you might look a little something like this. Right? If you look a little something like this, to get to this type of excessive size, in this case, there is actually some new muscle cells that are being produced. Hyperplasia does have to take place to get to this level of growth. And again, you see that in people, for instance, like Arnold Schwarzenegger. Arnold Schwarzenegger looked like this and more. There you go. We were just talking about the Terminator, like this and more in his heyday. But once he stopped working out regularly and got into politics, he shrank dramatically. However, did he shrink back down to the size of a normal individual at that point? No, because he still has more muscle cells. His muscle cells got smaller, but they didn't go away entirely. And there you go. That's the other key to this as well. If we all worked out, did the exact same number of reps with the exact same number of weight, the exact same number of times a day, we would all not gain the same amount of size. There is a very strong genetic component to how big and how strong that we can get. So absolutely. And no, it does not necessarily take human growth hormones or other types of anabolic steroids to get to this size. That certainly helps, makes it occur faster, but this can be done without that as well. Oops, didn't mean to do that. All right. So, is it possible to make new cells? Yes, but it's not the normal way that we grow. All right, questions on that? Protein shakes help. If you're going to make a lot more proteins, if you're going to make a lot more muscles and muscle cells, then yeah, definitely having the proteins for that helps. Yeah. All righty. Our next goal is to take our single muscle cell, that single muscle fiber, and look at its specializations. All right. Again, we know the basic anatomy of a cell. We've learned that already. However, as we've also learned, as things become specialized, we are going to give them special names. And we see that starting first with our muscle cell. We know our muscle cell, let's go ahead and make it black. Is a big, long cylinder shape. And as we know, like all cells, it has a plasma membrane. And inside of that plasma membrane, we have the cytoplasm. Right? Those are not new terms. That's nothing new. That's nothing surprising. We've had all of that before. However, our plasma membrane of our muscle cell is highly specialized. So the plasma membrane is highly specialized to produce and propagate. What does it mean to propagate?
To take like a small piece of something and grow a new one, like plants. I know you don't like plants.
That's well, I mean, it's not like I don't not like them. It's not like chemistry, right? But I'm just saying that they're stupid. That's all. No, I actually, I'm just saying I know nothing about them. I know absolutely nothing about them. I am a human. I am an animal person. My knowledge. But you have absolutely the right idea. That is an example of propagation. Propagation just basically means to spread, right? That's basically what it means. So what it's able to do. Oops, that's not how you spell highly. Highly specialized to produce and propagate, which again is just a fancy way of saying spread, and action potential. Oh, they're pretty. I just know nothing about them. Again, I'm not disparaging plants. I'm just saying I know absolutely nothing about them.
No, it was like a TikTok meme. That's why I said it.
Okay. Not familiar with that one. Sorry, I'm old. All right, excellent. So this highly specialized plasma membrane that is capable of producing and spreading an action potential. And so as a result of it, we give it a special name. And the special name for the plasma membrane of our muscle cell is the sarcolemma. So sarcolemma is the special name for our plasma membrane of our muscle cell. The cytoplasm is also very specialized, has some specialized components to it. So it gets a special name as well. We call it the sarcoplasm because it's highly specialized as well. And as you know, sarcoplasm is made up of the fluid component, the cytosol, and is composed of organelles. And the sarcoplasm is specialized in both ways. In our cytosol, we have inclusions. Remember, inclusions we talked about when we talked about cells are special components, special chemicals that are found in just one or a few locations. One of the inclusions found in our sarcoplasm is a massive amount of glycogen. Someone remind me what glycogen is again?
Sugar.
Absolutely, sugar. And why would we want massive amounts of stored glucose in there?
Energy.
Energy, right? And how do we break down that completely to get all of the energy we need out of it?
ATP.
Well, ATP is what we need, but how do we break down the glycogen, the glucose completely to get as much ATP out of it as we can?
Is it substrate level phosphorylation?
Well, wasn't there some type of organelle that allowed us to completely break down the glucose?
Oh, is it the it's not the sarcoplasmic reticulum?
Nope, we'll get to that one in a second.
Mitochondria.
Mitochondria, there you go. Excellent. So we have a massive amount of glycogen for that fuel, and we need mitochondria to break it down efficiently to make ATP. However, if I remember correctly, my mitochondria need something to be able to completely break down glucose efficiently and extract all of that ATP. What does it need a lot of?
Is it calcium ions?
Well, calcium makes cells do wonky things, and we'll get to the calcium in just a minute. But what do our mitochondria need so that we can... Oxygen, absolutely. We need lots and lots of oxygen. And so another inclusion inside of our sarcoplasm is a special protein, and that special protein is called myoglobin. Myoglobin, notice, sounds an awful lot like hemoglobin. And myoglobin's job is to store oxygen inside of the cell. So notice we have massive amounts of sugar, massive amounts of oxygen, and massive amounts of mitochondria, all to do, efficiently make the ATP to do the work that this cell needs to do.
Never mind, you already wrote it.
Okay. Now, someone also mentioned calcium. And yes, contracting a muscle cell is a very, very wonky thing. And that wonky thing requires calcium. So we don't want to rely, even though there's a lot of calcium outside of cells, we don't want to rely on that. We want calcium inside the cell ready to do work for us. So luckily, our cell, our muscle cell, has a very enlarged, very specialized smooth ER that we call the sarcoplasmic reticulum. And its job is to store and release calcium. Remember, we said 99% of the calcium in the body is stored in the matrix of our bones. But outside the matrix of our bones, this sarcoplasmic reticulum inside of our muscle cell has the second highest amount of calcium. So it's matrix of the bones, and then the sarcoplasmic reticulum of our skeletal muscle cells is where we find the second most amount of calcium in our body. So massive, massive amounts of this calcium is stored inside the sarcoplasmic reticulum. And then lastly, we need one more very, very special organelle, and that is going to be our contractile organelle, which we'll talk about in a minute, but I will tell you now the name of it is called a myofibril. So that myofibril is our contractile organelle. Now, there's a little bit more organization we have to talk about. Again, we'll talk more about this myofibril in a minute. So for now, I'm just going to draw it as a green. That works. Just throw a couple of those in there like that. A long green thread-like elongated organelle that runs the length of the muscle cell. Because there's a couple other interesting things that happen here in the anatomy of our, and again, I guess we should probably write this up at the top as a reminder. We're looking at the anatomy of a single skeletal muscle cell. No. Black. One of the interesting things that happens is that our sarcolemma the plasma membrane that covers the entire surface. All right, hold on. I just saw that there was a big text here. So yes, absolutely. Remember, now we are focused solely on skeletal muscle cells. So we're just talking about skeletal muscle cells. We'll talk about how this relates to smooth muscle and cardiac muscle at the end. But right now, yes, we are solely talking about skeletal muscle cells so far. So now that being said, the plasma membrane of all muscle cells are called sarcolemmas. So again, because it has to do with the muscle. But right now, yes, we're focusing on skeletal muscle. What's interesting about these, the sarcolemma of our skeletal muscle cell is that it actually has these invaginations, these folds in the plasma membrane that penetrate deep down into the muscle cell and actually wrap around. Oh, I think I can actually do this. If I move this up to here, that's going to be, no, there you go, perfect. And then I can cheat and do that. Hey, there you go. It penetrates deep into our muscle cell, wraps around our myofibrils, and I guess I should name this. So there's our myofibril. This fold in the plasma membrane that travels down into the muscle cell, we call a transverse tubule. And again, make sure you call it the transverse tubule. However, if you're explaining this in an essay question, once you've written it out once, then you are allowed to abbreviate it as a T tubule. But don't use T tubule the first time you identify this on an exam or something. That is considered a spelling error. Remember, no abbreviations. And if you think about it, this transverse tubule is a fold of the plasma membrane. So inside of it, it contains interstitial fluid. So the same interstitial fluid that is located out here outside the cell is also located in our transverse tubule as well. Now, we just talked about the sarcoplasmic reticulum. The sarcoplasmic reticulum also wraps around in kind of this mesh-like structure, the myofibril as well. So let's label this as our sarcoplasmic reticulum. And again, if you don't like my drawings, we've got the pretty pictures from the textbook that I will show you in just a minute. But there is something really interesting about this sarcoplasmic reticulum. When it gets close to the transverse tubule, it enlarges, forming this big enlarged structure. And this big enlarged structure is what is known as the terminal cisterna. So our terminal cisterna are these enlarged ends of the sarcoplasmic reticulum. And notice the same thing happens on this side as well. I've got my sarcoplasmic reticulum wrapping around the myofibril. And at the end, it enlarges to form that big terminal cisterna. So notice what ends up happening here. In this region, we have two terminal cisternae. And our T tubule. And these three things collectively are known as the triad. So we have this triad, which is the two terminal cisternae and the transverse tubule together, all wrapping around that myofibril. Notice we go to our illustration here. Here we see the pretty picture from your textbook that shows this. I'll use my I won't use my highlighter. Notice here's our sarcolemma, and notice the little holes are the folds where it invaginates and folds into, into the cell, wrapping around the long thread-like myofibrils. Yes, Yulia.
So is the purpose of this transverse tubule to bring nutrients or no?
Nope. Let's identify the structures. Once we identify all the structures, then we'll be able to talk about the function. But to understand how it works, we have to know all the pieces first.
Okay.
Absolutely have the right idea. The whole point of us taking it and breaking this down all the way to the protein level is so that we'll able to understand how all the pieces work. And then we can start building up till we understand how we contract the muscle. That's our goal. Our goal is to understand what just happened right there, how I contracted that muscle. But to do that, we have to know all the bits and pieces. And once we know all the bits and pieces, we can understand how they work together. All right. So like I said, we have a specialized plasma membrane called the sarcolemma, specialized cytoplasm called the sarcoplasm, containing those glycogen, myoglobin, large amounts of mitochondria. That's these purple things that we're seeing in our picture here. Those folds here in yellow are transverse tubules penetrating deep all the way to the center of our muscle cell. And again, wrapping around those myofibrils, containing that interstitial fluid. And here in blue, we have our sarcoplasmic reticulum, storing that calcium. That is specialized smooth ER. However, notice at the ends where it meets the transverse tubule, it enlarges to form those terminal cisternae, or terminal cisterns. And the two terminal cisternae and the transverse tubule collectively, these things together form the triad. Notice, again, to fully beat this dead horse, which I absolutely positively want to do, if we come back to our model like this, notice we're seeing the same thing. Notice here in orange is our transverse tubule coming in from the plasma membrane. Here in yellow, pardon me, here in white is our sarcoplasmic reticulum. Notice it enlarges at the end, forming the terminal cistern, and the two terminal cisternae and the transverse tubule form the triad. Now notice they've given us our mitochondria in here as well. Are our mitochondria all going to line up like ducks in a row like that inside of the actual cell? No, of course not. But again, it's just that artistic license. And notice we even see the same thing on this model. Again, notice here the blue is the transverse tubules coming in. Here, the light beige is our sarcoplasmic reticulum. And here we see the triad made up of the two terminal cisternae and the one transverse tubule. So again, notice we're looking in both of these models, we are looking inside of a single muscle cell. All right. Questions on that? Again, this isn't making sense. It's just going to get worse from here. So any questions?
Yeah, I'm still, so everything that's blue is this sarcoplasmic reticulum.
Correct.
And it's from my understanding, it surrounds a single cell.
No, this is all inside of a single cell. These long structures you see here. So again, here, let's cheat and put stars on them. This, that stars don't show up well at all. Do hearts show better? Sort of. Every single one of these things, I'm just cheating and using my stamps, is this organelle known as a myofibril. Long thread-like organelles that form both the structure and the function This organelle is the structural and functional unit of the muscle cell. These are the organelles that make it work. So notice the transverse tubules, the sarcoplasmic reticulum are wrapping around the myofibrils. But this is all just one muscle cell we're looking at. Well, the only thing that it was, I only have a few stamps. See, I got a check mark, I've got an X, I've got, what's the other ones? The star. Ooh, question mark. I like the question mark. So yeah, so I've got a couple cheat stamps. So just the heart seemed to make sense. All right, because it was easy to see. But that's the point. All of these things are myofibrils. They are the structural and functional unit of the muscle cell. They are what allow the muscle cell to contract, and they're made up of proteins. And the precise arrangement of those proteins is what gives the muscle its stripes. Okay? So, these proteins, as someone mentioned earlier, are what are also known as the thick and thin filaments. And because they're proteins in a muscle cell, they are known as myofilaments, right? Because myo means muscle. So there are these massively precisely arranged proteins that form all of these organelles that are the myofibrils. All right. So here we have my simple illustration that shows these internal structures. We just looked at the pretty picture from the textbook. Any questions on this? Because what we need to do now is, or next I should say, is we now need to look at the organization and anatomy. I don't know why everything's in caps. In the myofibril. So that's what we're going to do next. This strand organelle. We are going to look at the proteins that are making it up and talk about them and talk about their functions and identify them. And that's going to be our next goal. So we're continuing to go even deeper. So now we're not even looking at a single muscle cell. We are going to be looking at a single organelle in a single skeletal muscle cell. But that's going to require a lot more. Oh, yes, Laura, you had a question?
Yeah. Just before we move on, I want to kind of make sure that I understand. So in the sarcoplasmic reticulum, the terminal cisterna is the, or I guess you could say swollen sarcoplasmic reticulum, but is it swollen because it has a lot of the calcium ions, or?
Absolutely.
Okay.
It is swollen because it contains more. So again, let's get away from my drawing and go back to the picture here. So yeah, notice that it's more thread-like in the middle, but at the end it enlarges. So absolutely, in that enlarged parts, it has more calcium in them. So notice there's going to be more calcium in these larger parts next to the transverse tubules.
Okay.
Absolutely.
Now, is it, I don't know if it's like that just because of how the structure is, but is there, kind of want to make sure I say it right. Is it calcium on the ends so it can, for structure, or is it just?
No, okay, no, it is, the sarcoplasmic reticulum is a balloon that contains the calcium.
Okay.
That is going to let it out when we need it to do its job, right? Calcium makes cells do wonky things, and contracting is definitely a wonky thing. However, do we want our muscles contracted all the time?
No.
No. So we don't want calcium rolling around freely inside of our muscle cell, because we don't want to just be contracting all the time or contracting randomly. We want to control when it contracts. So when we want it to contract, we can let that calcium out, have it do its job, and then lock it back up when we need it, right? Folsom has one of the highest concentration of criminals of any state in California. Does that mean you shouldn't live in Folsom?
No.
No. Why? Where are all the criminals?
In the jail.
In the
Locked up
prison.
in jail, exactly. They're locked up in jail where they can't cause any harm. And it's the same thing here. The sarcoplasmic reticulum is calcium jail.
Okay.
Of course, when we want the muscle to contract, then we have to have a jail break. And all that calcium has to rush out. We can get it to do work, and then we can slam it back in when it's time. All right?
Thank you.
Arthur, you had a question?
Yeah, can you explain the difference between terminal cisterna and triad again?
Yes. And I'll just do a real simple drawing here. Our sarcoplasmic reticulum, like we just finished talking about, is the smooth endoplasmic reticulum. It is a flattened membranous sac that contains calcium inside of it, stores calcium. However, At the end of it, it enlarges and it gets bigger, which, as was pointed out, allows it to store even more calcium inside of it. That enlarged portion of the sarcoplasmic reticulum. So, and here I'll use my highlighter for this one. This enlarged portion right here is a terminal cistern. And there's a terminal cistern, and there's a terminal cistern, and there's a terminal cistern, and there's one, and there's one, and there's one, and so on and so forth. If you notice, 2 terminal cisterns surround a single transverse tubule. So these 3 structures together, the 2 terminal cisternae and the transverse tubule, those 3 things together are the triad, right? Triad, tripod, triceratops, tricycle. Tri means 3. So this triad, the 3 things coming together are the triad, the 2 terminal cisternae and the transverse tubule in between, right? Like an Oreo.
So the 2 terminal cisternaes and the tubules, they help store the rest of the calcium ions.
Transverse tubule does not store calcium ions. The calcium is located in the sarcoplasmic reticulum, and there's a lot of it in the terminal cisternae. And we'll see. So here, if we look at it from a side view, our transverse tubule comes down from the top of the plasma membrane.
Mm-hmm.
And along the side, we have our 2 terminal cisternae, which have a lot of calcium inside of them, sitting right next to the to the transverse tubule. So this has calcium in it, this has calcium in it. But remember, our transverse tubule has interstitial fluid inside of it. So it's the slightly different composition to it there. This is outside the cell, whereas obviously this is inside. All right.
Making sense now. Okay.
Yep. And again, right now we're just focusing on the structures, and then we'll talk about how they're all functioning, how they work together, and how they do their job. So we will get to the function of them, but right now we're just identifying all of these structures. All right. So what we need to identify next is what's going on with this myofibril. What is its anatomy? What are the proteins that are making it up? How does it do its thing? And so that's what we're doing next. But that's, again, going to involve a lot more vocabulary. So let's go ahead and take a second break. So for this break, again, we'll take another 15-minute break. So it looks like that means coming back at 3:08. We will restart, and I will start the recording at that point. All right. Questions on that? All right. I will see you guys in 15 minutes. Let's go ahead and get started. Our goal now is to talk about this myofibril, this specialized organelle. And again, as I emphasize, this is the contractile organelle. It is what is responsible for producing the tension, changing the shape of the muscle cell, and generating the force that is going to move our body through space. One of the nice things about it is it's very much like a train, right? If you've ever gotten stopped by a train before and you watch that train go by, a train is a singular thing, but really it's made up of a bunch of repetitive compartments, right? Especially if it's like a grain train, right? You could sit there for a half an hour while 100 identical grain cars go flowing by. And that's basically what our myofibril is. Our myofibril is this big, long, thread-like contractile organelle that is made up of a repetitive pattern of proteins. This repetitive pattern of precisely arranged proteins basically makes up the myofibril, and each one of these individual units is what is known as a sarcomere. So these sarcomeres make up our organizing pattern that is this contractile organelle that is our myofibril. So again, it contains some proteins forming a structure called a sarcomere. So let's take a look at this. Let's actually come back to the whiteboard. Again, I'll save this picture. Our goal now is to pull out this structure, this myofibril, and look at it. And so to do that, I'm going to go ahead and cheat and erase this and start my drawing from new. And again, at this point, we are looking at the organization of proteins that form a single organelle the myofibril. And again, which is comprised of the repetitive patterns of sarcomeres. Now, as I also mentioned, this is comprised of proteins. So we need to identify the types of proteins that are involved in this process. And again, because these are proteins found inside of a muscle cell, we can give them a fancy word and call them myofilaments. Now, there are three main types of proteins that are formed in this. And again, I will say that there are dozens of proteins that make up and are associated with the myofibril. I'm not holding you responsible for every single one of the literally dozens of proteins that make them up, but we are going to hit on the 7 most common and most important for our understanding of it. And those 7 myofilaments fall into one of 3 categories. The first category, and hopefully the most obvious category, are the contractile filaments. Guess what contractile filaments do?
Contract the muscles?
Yeah, their job is to produce the force or the tension, we could think of it, that changes the shape of the muscle cell. Excellent. So those are contractile filaments, myofilaments, and it turns out there are 2 of them. The first one is a contractile filament known as myosin. Oops. Myosin is a dimer. What does it mean to be a dimer?
For some reason, I thought dimers is what forms when there's DNA damage.
Nope. What? Anyone know what a
Is it a
dimer?
molecule consisting of 2 identical molecules?
There you go. Exactly. It is basically a molecule made up of 2 identical molecules. So basically, a single myosin subunit has 2 parts to it. It has a head-like region, and then it has a tail-like region. And so that is a single myosin subunit. However, as you pointed out, they come in pairs. So what happens is there's going to be another one here, and it's supposed to be identical, but let's pretend that it is. And then it connects here. And again, I'm going to have to cheat. These intertwine with each other, so I'll just wrap it around it here like this. And so basically it has a tail region and it has a head region. And so this is what basically a single myosin myofilament looks like. Now, what's special about the myosin is, as we talked about, the myosin head is a motor. And what did we say a motor does again?
Takes chemical energy to mechanical energy.
Exactly. So what it does is it basically breaks down ATP. I don't know if you guys can hear my dog going crazy in the backyard, but it's pretty funny. Breaks down ATP. And when it breaks down ATP, what it does is it actually causes the myosin head to change position. It causes it to pivot, where basically it pulls forward. So essentially, it grabs and pulls. All right. It's holding on to something, and with ATP, it pulls. That's basically what it does. It converts that chemical energy into mechanical energy. Now, notice I'm sitting here grabbing and pulling. Am I going anywhere?
No.
Why not?
Because you're
Why
not
aren't I?
attached to anything.
Right, because there's nothing for me to grab and pull on. If for me to move, I would need to actually be holding on to something and actually grabbing and pulling. And that's what myosin needs. And that's what our second contractile protein is. Our second contractile protein is your friend and mine, actin. Actin, remember, is also a dimer comprised of 2 globular subunits. So, oops, no, let's use a different color. So there's one and there's one. So again, it's just 2 of these together. And the nice thing about this organization is it's very simple. So we can stick a couple of them together and then 2 more together and then 2 more together. And suddenly I have a whole actin filament. And on these actin dimers, they have a binding site, a handhold that allows myosin to grab onto it. So that's basically what happens. Myosin grabs onto the actin and pulls, and as that pulls, that's generating the force. That is generating the tension. All right? So these are our 2 proteins working together, myosin and actin. I'm going to cheat here. And it has a myosin binding site, or what is also called the active site. Either of those are acceptable terms that our myosin is able to grab onto and pull. Hopefully that's pretty straightforward, close enough. Questions on that?
So where is the myosin's active site? Where does it connect?
So basically the head of it has a binding site on it where it's actually able to grab onto the actin. So this binding site binds to that binding site. And then when it does that, it's able to undergo that conformational change, do that pivot, and generate that force. Okay? And that's basically what happens. Myosin pulls on actin, and that's how we move our body through space. Now, myosin really likes to pull on actin. And as long as there's fuel, ATP around, it'll do that. It'll want to do that. And how often is there ATP in your muscle cells?
Yeah, often.
How about all the time? Absolutely. So why aren't your muscles contracted all the time?
They're
Because
always actively holding hands.
Well, are they? Are your legs right now actively contracting? Is there a lot of force being generated by your legs right now?
No.
Probably not, right? So those muscles aren't contracting really. So how is it that if myosin always wants to grab and pull on actin and always wants to contract the muscles, your muscles aren't constantly being contracted all the time?
there's like this one thing on the binding sites that prevents it from
Exactly. And there's in fact
keeps it
2
closed or
things.
something.
And those 2 things are our regulatory proteins. You are absolutely correct. We have these regulatory proteins, regulatory filaments, and there again are 2 of them. And it is their job to basically turn the contraction on and off. Sliding filament, well, these are filaments, so we're going to need to get them to slide. But again, to understand the physiology, we need to know all the pieces first. And so we will see how this comes into it. But you've absolutely got the right idea. If myosin and actin are going to grab, and if myosin's going to grab and pull on actin, those filaments are going to slide, and that's exactly what's going to happen. All right, now, myosin wants to grab onto actin and wants to do it all the time. But luckily for us, there are these regulatory proteins. One of these regulatory proteins is a big, long, thread-like protein that sits on top of the actin. And this protein is what is called myomycin. Pardon me, no. Tropomyosin. Tropomyosin. So our first regulatory protein is tropomyosin. It's a long thread-like protein, and it covers the active sites on the actin. So notice, with that thread now in the way, myosin can't grab onto the actin, it can't pull, it can't produce any tension. However, eventually we are going to want this this muscle to be able to contract. So we need to have a way to move this tropomyosin out of the way. And luckily for us, we do. Sitting on top of the troponin is this handle-like protein, and this handle-like protein is called troponin. Troponin is a handle-like protein. That can rotate the tropomyosin out of the way. And of course, if I'm able to move that tropomyosin out of the way, that will allow the myosin and the actin to interact. So notice, this is going to allow me to turn the contraction on and off. When I want the contraction to occur, all I have to do is move the tropomyosin. When I want the contraction to stop, all I have to do is move the tropomyosin. So by moving that tropomyosin over and away from the binding site, I can turn the contraction on and off.
The troponin is the blue rectangles you drew?
Yeah, the blue rectangles are the troponin. Those are the handles that are moving this thing out of the way. And again, I appreciate my art isn't the greatest, so let's take a look at some pretty pictures from your textbook. Here, notice we have our myosin. Here we have a single myosin dimer with the heads and the tails. Remember, as I mentioned, the head is the motor, converts chemical energy into mechanical energy, allowing it to pivot. Here we see a couple things. We see the individual actin dimers, and notice they have that active site, that binding site for the myosin head on it. But notice here we see that thread-like tropomyosin over the binding site and the handle-like troponins that can move it out of the way. Tropomyosin troponin and tropomyosin, these regulatory proteins, control when the contraction occurs. So notice they're not producing any of the force. They're not generating the tension. That is what our contractile proteins are doing. But they control when our contractile proteins can interact. So notice, like on this picture here, right, the myosin couldn't grab onto that actin because all of our binding sites are exposed. Are covered. But if we moved them out of the way so they were exposed, then it would be able to contract. So they don't help in the contraction. They don't generate any of the force. They just turn it on and off. And that's the key, right? So if you notice, we have two types of proteins so far. Contractile proteins, these are the ones that generate the force. These are the ones that produce the movement. And regulatory proteins that control it. They turn it on and off. All right. Questions on that? All right. Now, obviously, these allow us to turn the contraction on and off. These allow us to generate the force. But can we throw these proteins just randomly into a cell and hope for the best?
No.
No, absolutely not. Remember, this is both the functional and the structural organelle. Not only does this allow the muscle cell to contract, but remember, this is what gives it its stripes. So our third type of proteins are what are known as structural proteins. And these structural proteins are what give the shape and the structure, the stripes to it, and allow this to be efficient. And again, I keep emphasizing the precision of this and the importance of this, right? But think of it this way. If I grabbed any four of you, even though all of you have your cameras off right now, I'm guessing most of you are pretty fit, pretty healthy, pretty strong individuals. So I could put four of you in a boat and you'd be able to row that boat from one side of a lake to another, right? You pretty comfortable with that idea? However, excellent. Yeah, absolutely. However, if I made you compete against a crew team, and assuming that crew team wasn't made up of Lori Lofton's kids, are you guys likely to beat that crew team to the other side of the lake? No, not because you're necessarily not as fit or not as strong or anything else, but they train, and one of the things they train for is their precision. They are all putting their oars into the water at the same time and all pulling at the same time and all rotating them at the same time and moving them at the same time. And that precision leads to an increase in efficiency, an increase in strength. And so that's what happens here. By having these things precisely arranged, that enhances the efficiency and makes the muscles stronger. So we need to have these precisely arranged. And because they're precisely arranged, they are going to be much more efficient and produce much more strength. All right. I think I can sneak this in down in the bottom here. Now, like I said, there are almost a dozen different structural proteins. I'm only going to hold you responsible for 3. All right. So let's start looking at a single sarcomere. With a single sarcomere, obviously it has boundaries. I have a room here, and this room has boundaries to it. However, one of the interesting things about this is I happen to have a wall right here, but if I were to knock on this wall or bang through this wall, where would I end up?
In the other
my house, you don't know, but my kid's room is right next door. So notice this wall we share. And that's kind of the same way with the sarcomere. The sarcomere has a boundary. One of these boundaries, what color haven't I used yet? Let's use purple. Is a zigzag protein known as a Z disc. And that Z disc forms the boundaries of my sarcomere. So from Z disc to Z disc is one sarcomere. But guess what? Over here, and I go, no, this isn't drawn to scale, but I'm going to erase it in a minute, is a second sarcomere. So notice this sarcomere is part of the wall of this one, and this Z disc is part of this sarcomere as well. So again, this Z disc forms the boundaries. And so let's write that. Z disc forms the boundaries of the sarcomere. Now, let's arrange some of the proteins we've already talked about. Starting first with the actin. Actin was red, so we'll stick with red. Actin actually attaches to the Z disc. It is anchored to the Z disc. So here we have our actin anchored to the Z disc. Our myosin, which we know is orange. We have one myosin here, but it turns out hundreds, if not thousands of myosins, we're getting to that right now, Ariana, of these myosins all come together. And when they come together, and I'm just going to draw lines here, I'm not going to bother drawing the heads. Hundreds upon thousands of these myosins all come together so that the myosin is actually what we call the thick filament. So you guys have talked a couple times about thick and thin filaments. Our myosin filament is the thick filament. And basically all these other filaments we've talked about, the actin, the Z disc, the other ones we're going to talk about, basically are the thin filaments. So myosin is our thick filament, and it is located here in the center of our sarcomere. And again, here I've just drawn one, but again, there's going to be another one down here, and it's going to have its heads coming off, attaching to the actin on this side, and so on and so forth. Again, I'm doing a little simplistic illustration of this, but hopefully you get the point. And of course, remember also we have to have our troponin and tropomyosin, our regulatory proteins there. So let's go ahead and put our troponins and a tropomyosin. I'll just put one on each of these to remind us that there are these handles that are going to move the troponins out of the way. Yes, Laura, you had a question?
Yeah, so on the slides that you showed us, you had mentioned that there were subtle like lines in between the thick and thin filaments. Are those the troponin and the tropomyosin?
Nope, but you are absolutely right. As we look at this, those subtle things in between the stripes are made up of these proteins, and we are absolutely going to see that. As soon as I'm done making this, we'll actually see where the stripes come from.
Okay.
Absolutely got the perfect idea. Absolutely, absolutely. Excellent. I like that. Perfect. So here are the things we've talked about so far, and so far so good. We've taken not just our proteins, but we've organized them and started organizing them into the sarcomere. But notice we need two more structural proteins. First, we need a way to stabilize this myosin in place. Notice the myosin isn't really being held by anything right now. So it would help if we had some additional structural proteins to do that. One of those structural proteins is a structural protein known as titin. Titin comes from the same root of the word titan. And anybody remember who the Titans were in Greek mythology? We've actually talked about one of them in this class. The Titans were the gods before the Greek gods. Yep, we're getting to where that is in just a minute. The Titans, remember we talked about Atlas, like our Atlas. Remember he was one of the Titans who was punished by Zeus and has to hold the head on his shoulders. Titans were big, massive beings. And this protein, when it was first discovered, was the largest protein that had been found in the body. Now, we have since found proteins bigger, but because of its status at the time, it got called titin because it was the largest protein. Yes, it starts in this coiled nature attached to the Z disc and then basically stabilizes the edge of the myosin. There will be another one on this side as well with that same type of organization. Now, notice this does a couple of things. One thing that these titin proteins do is to indeed stabilize the myosin, helping to hold it in place. But notice with the coiled ends, kind of like a slinky, if you take a slinky and elongate it, do you damage that slinky?
No.
No, absolutely. Notice you can stretch it without damage. Extensibility. And as Laura pointed out, if you let go of that slinky, what happens to it?
Pulls it back together.
Goes back to its original shape. So not only does this help to stabilize the myosin, but this titin is what provides the extensibility and the elasticity. It allows it to be stretched without damage, and it allows it to recoil back to its original position. Really, really important in its function that way. There is one final structural protein, because notice we saw that not only is this these proteins in this one sarcomere in this one myofibril precisely arranged, but remember, it's got to be arranged with the myofibril next to it, and the myofibril next to that, and the myofibril next to that, and even the myofibrils in the next cell. So what we need is we need some additional proteins. Oops, no, hold on. I want that to be there. And then the last protein is a protein called myomycin. Myomycin is this long thread-like protein that anchors all of the myosins from all of the myofibrils together and attaches it even to the plasma membrane of the wall. And in fact, I have to cheat a little bit here. The titin attaches to it as well. So the titin attaches to it to this side, titin attaches to it that side. And these help to organize not just this myofibril, but all the myofibrils next to it as well. So again, my simplistic drawings have done this, but let's look at the pretty pictures from our textbook, and let's go back. As I mentioned, there are three types of proteins: contractile proteins that generate the force. And remember, there are two of those, myosin and actin. There are regulatory proteins, the ones that turn them on and off. There are two of those, troponin and tropomyosin. And three regulatory proteins: Z discs, myomycin, and titin. And they maintain the organization, maintain the efficiency of these. And we've got lots of pretty pictures that show this. Here we see that big, thick myosin filament with hundreds of myosin heads coming off from all of those dimers going together. And again, as we mentioned, this forms our thick filament. Its head is that motor, converts ATP into mechanical energy, allowing it to pivot, allowing it to pull. And what does it pull on? It pulls on those actin dimers with their active sites, with their binding sites. I love this picture from your textbook. Here, this is an electron microscopy view. Using an electron microscope, we're able to see all of the organization of this. Notice we have these big, huge, thick myosin filaments here and here and here. And notice coming off of them, we see all the myosin heads that are coming off of them. And this one's coming off this one here, and this one there, and this one there, and this one there. And notice how they're connecting to the actin in between. So there's the actin, and there's our actin, and there's our actin. So you can see how they're overlapping with each other, and not just in 2 dimensions, but truly in 3 dimensions. In fact, if we cheat and go back to that muscle model handout I had, notice there is actually a model of a sarcomere. Notice here it's got the plastic wrapping around it, representing the transverse tubule. And the green here would be the sarcoplasmic reticulum with the terminal cisternae. So notice we have a triad here. But if we peel that off, and there we see it here, we can see, well, let's start here, a sarcomere from Z disc to Z disc. Notice here is the Z disc. Notice here in the middle is the thick filaments, the myosin heads on them. Here are the thin filaments, the actin. Notice in yellow, we see the tropomyosin, and the little green are the handles that move it, the troponin. Notice here's that myomycin on the midline, stabilizing all the myosins together in this sarcomere, and also with the next one that would be there as well. The only thing that's really missing from this model is it doesn't have the titin in it. So we have a great model in the classroom, which absolutely you need to know for the exam. But we also have Where'd it go? Oh, there's the slideshow. We see here this with the electron microscope, but again, here we see the troponin and the tropomyosin. And here we see a simple illustration of a sarcomere. Again, Z disc to Z disc, but notice actin attached on this side, actin attached on that side. So this Z disc is the boundary of this sarcomere and also the boundary of the next one. I see your hand, Arthur. Let me just finish this real quick. We see the actin, we see the myosin with its thick filaments. Notice this one doesn't show us the regulatory proteins that would be there, but it does show the titin. It shows the myomesin, although it shows it as an anchor. Really, it's 3 strands of that, and then the Z disc as well. Yes, Arthur, your question.
Yeah, I was looking up stuff like back when I was doing my pre-lab or whatever for the homework for today. And I was looking some stuff up and I found that this is actually called like a sliding filament theory. So is it a theory or is it something that was considered a theory back then?
Well, so again, gravity is a theory. From a scientific standpoint, a theory is just something that hasn't been 100% proven. But just because gravity hasn't been 100% proven doesn't mean I'm jumping out my window anytime soon. And it's the same thing here. Remember, we're looking at something at a microscopic level. That's something that takes an electron microscope to see these proteins. And again, think about what you have to do with an electron microscope. First, you have to get a piece of tissue, dead tissue, freeze it, slice off the tiniest, like 30 micron slice of it, and then you're bombarding it with electrons. Clearly, that's nothing we could do with living tissue. So has anybody ever actually observed these actual myosin heads grabbing and pulling on the actin? No. We understand the mechanisms, we understand how it should work. And so again, from a scientific standpoint, it's a theory because it hasn't been actually observed, like physically observed that way. But yes, these are the proteins involved in that sliding filament theory. However, if you prefer, and we'll also use the term the contractile cycle. So contractile cycle, sliding filament theory, it's the same thing. You're right. It's how these things are going to interact.
Okay.
All right. Excellent. Great question. So myomesin is the protein. Z disc is the protein. But as we will see, these things arrange to form our stripes. And the stripes are things that are like bands and lines. So you're correct. Myomesin is the protein that forms the M line. The Z disc is the protein that forms the Z line. Myosin is the protein that forms the A band, and so on and so forth. So yes, they're the proteins, but they're arranged in a precise fashion. And that precise fashion that they're arranged are all things like the lines and bands. And we're definitely going there next. All right. Again, here's another nice illustration that shows this. Again, it doesn't show the regulatory proteins on it, so we don't see those there, but we see the myosin in its head, we see the actin, we see the titin, we see the myomesin, we see the Z disc, we see all of those. And to get to where you guys are talking about, this arrangement, this precise arrangement of these give us our stripes. Now, these stripes are known as the A band and the I band because of the German words that were used to describe them when they were first discovered. However, what I find for me incredibly convenient is that dark has an A in it and light has an I in it. I want to emphasize that is not how they got their names, but it is an easy way to tell which is which. Now, the M line, pardon me, the myosin, which is the thick band, that thick filament, is the much denser protein structure. So basically, the portion of the sarcomere that has the myosin in it is what we call the A band. So A band is defined as the region that contains Myosin. So guess how we define the I band?
A region that doesn't have myosin.
There you go. Not a trick question. Just that simple. The I band is the region that does not contain myosin. The A band is the region that does contain myosin. Laura, yes, go ahead.
Now you said the I band is the region that does not contain myosin. Is that the only one that contains actin, or am I?
No. So let's take a look at this. Although I love where you're going with this. So again, we have to cheat a little bit. So let's draw a little bit. So we know this titin over here is going to connect to some myosin. That's going to go off that way off screen. So notice then basically this would be our I band, this would be our A band, and this would be the next A band coming off of it. So we've got this repetitive pattern, dark stripe, light stripe, dark stripe, light stripe across the entire cell. So that makes sense so far?
Yeah.
Notice we only use myosin in the definition because notice myosin is only in the A band. But notice actin is in the I band and the A band. So we can't use actin to define it because it's in both.
Okay.
Now, getting back, remember when we looked closely at our light band, which we now know is the I band, remember we saw a dark line down the center of it. That dark line in the center of the I band is what is known as the Z line, formed by the Z disc. Because notice our A band crosses from one sarcomere to another. So our A band crosses sarcomeres, whereas our A band is completely contained in one sarcomere. All right. But you've got you're making a great point with that actin. Notice if we look closely at our sarcomere, and I'll use dark black to emphasize this. For a lot of our A band, myosin and actin overlap. But as we know, most is not all. There is a small space in the center of the A band that does not contain actin, only contains myosin.
Okay.
And this region that only contains myosin, we call the H band or the H zone.
So the band that only contains myosin is the H band, but the M line is what holds both.
Yep. So notice H band is in the center of the A band. So the H band only contains myosin and it's in the center of the A band. And you're absolutely correct. Notice in the dead center, in the A band, we have both myosin and myomesin. And this area dead center that contains myosin and myomesin, we call the M line. And that is this region right here. I'll color it yellow, right in the center. So notice we have A bands, I bands, M lines, H bands, right? And a Z line. All this different organization. We can see it on an illustration like this. Notice if we go back, heck, we can cheat and look at it here as well. Notice when we look here, M line. Notice this part here would represent the H zone. Right, here's just the A band that contains all of the myosin. And here we would see the little bit of the I band that does not contain myosin. Although where I think we'll see it nicest is if we come back to our illustration here. Oh, actually, no, hold on. I lied. I want this model. Notice when we look closely at this model of the muscle cell, we can actually see they've done a really nice job of showing us our proteins. Notice here in pink are those precisely arranged Z discs. So basically from here to here is a single sarcomere. Notice at the midline, we have our 3 myomesin protein fibers stabilizing the individual myosins that we see here. And notice coming off of our Z disc, we have our actins. So we see that nice organization. And what's cool, what's really, really cool about this model is you can see that it's cut the different myofibrils at different heights, at different regions of our organelle. So let's look at that. If we notice, if we look at it from the top view instead, which is why we have this view here, we can actually see the different regions of the myofibril organelle. So let's think about this. Notice in this section right here, we can see we have cut through both the thick filament and the thin filament. So because we've gone through both thick and thin filament, the actin and the myosin are overlapping. What region of the organelle would this be?
The A band.
A band, exactly. However, notice right here, we have just the thin filaments, just the actin. And so if we have just the actin, what region of the organelle would this be?
The I band.
Excellent. Notice this one here. Oops. Just thick filaments, just myosin. Oops, I didn't want to use orange. I haven't brown. I want to use orange. This one just has the thick filaments. And if it just has the myosin, what region would this be?
The H zone.
H band, excellent. Or again, you can use H zone. I'm fine with either. Both are acceptable. And notice, lastly, right here, we have myosin that is interconnected by the myomesin, locking them all in place. So this region where we have both myosin and myomesin, what would this region be?
M-line.
M-line.
Oh.
There you go. See how easy that is? And that's what they've done in the illustration from your textbook as well. Notice in the illustration from your textbook, this first section is right through the I band, where there's just the actin. Yes, titin's in there as well, but you're not going to see it. Whereas if you have the two of them overlapping, that would be the A band. If you just had myosin, that would be the H band. And if you had the myosin and the actin the myosin and the myomesin overlapping, you would be on the M line. So notice here what they've done in our illustration. Our model has actually done physically. Cut our myofibrils at different levels so we can see all of the different regions of the organelle and how this thing goes together. Awesome. Questions on that? Now, obviously, we're looking at all these pictures, and these are all illustrations, and these are all drawings, and it can't really look like that, can it? right? Here again, we see this nice illustration. Well, guess what? It really can. Using an electron microscope, here they are showing us with that electron microscope a portion of an actual myofibril. Notice, and I love this picture. There's so much that I love about this picture. Notice right here in the center, you can see where it's kind of sloppy. And here it's kind of sloppy as well. This is, well, the good news is we're almost done. And then you have a whole 2 days to synthesize this information. This is the boundary of the myofibril. So this is where the transverse tubule and the sarcoplasmic reticulum is wrapping around that myofibril. Notice within this myofibril, we can see the Z line to Z line. So here we have one sarcomere. We can clearly see the I bands that don't contain myosin and the A bands that do. So notice the myosin is completely contained within the sarcomere, whereas the I band crosses from one sarcomere to the next, which is why the Z line is right in the center of it. But notice also, and I'll clear this for a second so we can see more closely. Notice right at the center, we can see an area that is slightly lighter in color. It is lighter in color because instead of myosin and actin overlapping, it just has myosin, except for right at the center where the myosin and the myomesin overlap, forming the M line. One of the cool things they can do with these electron microscopes is they can colorize it. Now notice again, here we see the boundary between two myofibrils. And remember we talked about total precision, but notice it doesn't totally line up perfectly, but really that's just because of what they've done to this. Remember, they had to freeze it, they had to cut it off, they had to slice it, they had to bombard it with electrons. And even with all that messing with it, it's still pretty precisely lined up. But again, we can clearly see the A band, we can clearly see the I band. At the center of the I band, we see the Z line. At the center of the A band, we see the H band, and we see the M line right down the middle. Yes, Laura?
Yeah, can you explain the sarcoma one more time? So is it just the center where the A band, H, and M line are?
No, a sarcomere, remember, is that functional unit. It's the grain car. It's the train car. It
Okay.
goes from Z disc to Z disc. So
Okay.
from Z disc to Z disc is one sarcomere.
Gotcha.
Okay. And let's go back to our original picture. So notice here we look at it with a light microscope. With a light microscope, now we can see there's a Z disc. There's a second Z disc. So basically that's one sarcomere from Z disc to Z disc. And then to the next one is a second sarcomere. And to the next one is a third sarcomere. Notice we can clearly see the Z disc because we know the Z disc is in the center of the I band. But notice also when we look closely at the A band, there's an area right in the middle that's a little lighter in color. That is the H band. Now, we're not at this magnification, you're not going to see the M line. So we know there is a slightly darker place right in the middle, but at this magnification, we're not going to see the M line. But we do see the H band. We do see the Z line. We clearly see the A band and the I band. And like I said, not only are all the hundreds of myofibrils perfectly aligned in the single cell, but thanks to structural proteins, they also perfectly align with the cell next to them as well. Like I said, Yulia, it's okay if you're overwhelmed. My goal in this class is to present you with the information. You shouldn't master the information by the time I'm done talking. I'm exposing you to this information, so in the 20, 30, 40, 90 hours you're spending at home studying, you've been exposed to these things to hopefully make some sense with it. So the goal isn't to have mastered this by the time I'm done talking. I'm exposing you to it so that you have the opportunity to master it on your own. That is the goal. All right. And so just that simply, we have taken an entire muscle like the bicep brachia and broken it down to its ittiest bittiest, tiniest bits, the proteins that make them up. And now that we know all the bits and pieces, we can take those bits and pieces and put them together to understand how they work to make the muscle contract. Yes, Arthur.
Well, this goes back to what you just taught us,
Yep.
but I don't know if you talked about it, but how many actin filaments are there to myosin filaments?
Great question. So typically, so if we were, well, if we were to take this and cut it and look at it in cross section, basically what happens is you've got, so here, let's cheat and use this page. So you've got a single actin, and around that single actin are typically going to be, I think it's 6 myosin, and then each individual myosin is surrounded. Oops, that's not gonna work. You get the general idea. 1, 2, 3, 4, 5. Close enough. You get the idea. So they're typically 6 around. So as they wrap around each other, I guess my scale is off on this, but you get the idea. So yeah, 1 is going to basically have 6 myosin wrapped around it, and those myosin are going to have 6 actin, and they overlap. So again, so that's going to go there, and that's going to go there, and then another one here, and another one here, and so on and so forth. So up there and there, something like that. There we go. There, that's a little better. So basically they interweave with each other this way. And again, I've done a horrible job of drawing this, but I think what shows it best is this sarcomere model. Notice the sarcomere model, you can see how they're all overlapping with each other. They wrap around them. They're surrounding them from different angles. I don't think I have a front-on view of that. Nope, I don't. So I don't really have a front-on view. And the problem with these models is these pieces break off all the time as well. So they're kind of damaged. There are several pieces of it on the floor and stuff. But you get the idea, how they wrap around and intertwine with each other. The electron microscopy might be the best that shows it. So I keep showing it as a like 1-to-2 ratio, but that's clearly not the case. So as we go back to this original, well, like these, like this one here, there shows the nice, so there you go. So here you see the arrangement of the 6 actin around the myosin and so forth like that. So you see them wrapping around each other that way. And then also we see it with this, where you can see them overlapping with each other, and that's occurring in 3 dimensions. All right.
Is there a reason why we only have 6, or is there like a more efficient way of doing it? And do we see that in like other animals or other athletes?
So great question. Other athletes, absolutely not. Other species, I don't believe so. I think those that have myosin and actin use a similar mechanism for the overlap in skeletal muscle. As we'll see in smooth muscle, for instance, doesn't have the striations. So if you don't have the striations, you have a different arrangement to the myosin and the actin. But in cardiac muscle and in skeletal muscle, where we have the striations, we have the myofibrils, we have the sarcomeres, I think 6 is the number that of it that's wrapped around. And again, why? I don't have a good reason for that. I mean, again, why isn't an easy question to answer, but that's just the way that it is. But no, again, well, athletes may have more muscle cells, you know, or they may be more efficient in their muscle activity, but they're not the composition of it is going to be the same.
I mean, are there like disorders where you would have more? Like I've heard like some people have like some supernatural powers and stuff like that, where they're born with like a special ability to, like, I don't know, lift more.
So I can't speak to superpowers. What I can say about that is every single one of us is capable of lifting and moving and jumping and doing more than we do, because we have a lot of cognitive barriers in the place that limit our ability to do this because we don't want to hurt ourselves. But there's plenty of instances where you hear instances of, little old ladies flipping cars off of babies to save their lives, right? So again, when in the right circumstances with the right motivation, we can overcome some of those cognitive barriers. And we'll talk about this a lot when we get to fatigue. Because again, if my wife is an endurance athlete, she's one of those crazy people who likes to run marathons. And one of the things, for those of you who are endurance athletes and know, when you're training for a marathon, do you actually ever run the 26.2 miles before the day of the marathon?
No.
No,
Say again?
no, usually you don't. You get close.
In fact, typically when training, you never go above 20 miles. Because something special happens when you hit that 20-mile mark. What's that magical thing that happens when you hit the 20-mile mark when you're endurance running?
Like your runner's high or something?
Well, right before the runner's high is what happens. You hit the wall, right? They talk about this cognitive thing that happens where you hit the wall, where you are certain with 100% certainty that if you take one more step, you are going to die. Of course, if you take that one more step, do you die?
No.
No, and then they take a second and they take a third, and you break through that wall. And often breaking through that wall, you get that summer's high as an example of that, runner's high as an example of that. That wall that you run into is a very strong cognitive process that stops you from overexerting your body and potentially damaging it. Now, getting back to the things you guys are talking about here, yes, absolutely. There are some genetic abnormalities of individuals who can have, like you said, hypermuscularization. The cattle are a great example. You see some of those really buff-looking cattle. Those can be seen there. So yeah, they are pretty gonzo. So yes, there are some degenerative disorders or other type of genetic abnormalities that can cause those types of things. But that doesn't mean that our professional athletes are, genetic mutants. It just means that they've, change more efficiently, and many of them may have a genetic predisposition that helps them to gain muscle and things like that. But again, you also have to think about the type of muscle. Both Usain Bolt and Meb are world-class athletes, right? But if you looked at them side by side, they look dramatically different. Because what does Usain Bolt do?
Run.
Well,
distance.
he sprints short distances. What does Meb do?
I don't know him. Probably long distance.
Yeah, he's a marathoner, right? Very famous American marathoner, came over from, I think, Ethiopia or something like that, and to the United States, became a U.S. citizen, is probably one of the best marathon runners in the United States. And again, they look dramatically different. And if Usain Bolt decided tomorrow that he wanted to run marathons, would he necessarily be efficient doing that? Like right away?
No.
Probably not.
He hasn't built his body for that. So yeah, there's going to be a lot more that goes into this, and we will talk about all of those things. But for right now, we need to understand, yowzers, we need to understand the basic anatomy of this so we can understand the basic function, because that's what we're going to be doing next. And this is the last thing I want to leave you with. So while you're looking at this material, you can start looking ahead. A muscle contraction, as I said, I think last week, is the longest physiological process we will talk about in this class. Doesn't mean it's the hardest, it's just the longest. And because it is such a long process, from the point of when I decide to contract a muscle and that muscle actually contracts, we are going to describe that whole entire process. And it's long. And because it's long, we are going to break it up into 3 more easily digestible pieces. And we're not doing it randomly. We're picking meaningful points to cut it up, but we're going to cut it up into 3 pieces. And I guarantee you that at least 2 essay questions will be on these 3 processes on the exam. So these three processes, I guarantee, will be responsible for at least 2 essay questions on the exam. So it is going to be important to make sure we understand that. So as you start getting comfortable with the anatomy, if you want to start looking ahead towards Thursday's lecture at these 3 processes, that's what we're going to start doing on Thursday. Now, the other thing we're going to start doing on Thursday is talking about and identifying the muscles of the body and their origins and their insertions and their actions. And you have a list to help you with that. If you've not done so already, print out that list. If you haven't, first and foremost, shame on you. And secondly, do so. The other thing you're going to want to print out hold on, dupe, modules. So here we see the list. You guys can see what I'm doing on the screen, right? Yes?
Yes.
Okay.
Yeah.
So here we have our gross anatomy. Notice we have muscles, we have origins, we have insertions, we have actions, we have all of that that you're going to need. But the other thing that you're going to need that is under modules is you are going to need this origins and insertions handout. Notice this has the bones on it, and what we are going to do together is we are going to draw on it. So we are going to draw, like I said, origins. So we can draw something red that is an origin. We can draw something blue that is an insertion. And we can understand how that works by seeing what joint it crosses and understanding how the origin and the insertion relate to each other. So you will be tested on this. Obviously, the majority of this exam, are we on time? Oh, we're good. I have time to talk about this. Let's go here then. If you have not used your MyLab and Mastering, first and foremost, shame on you. But secondly, remember, go to your study area. And in your study area, go to your practice anatomy lab. And when you go to your practice anatomy lab, while it's got all these fun, cool cadaver stuff, we also have these anatomical models. This is really useful for when you're in the classroom, because when you happen to look at, for instance, the upper limb model, this is an exact replica of the model we have in the classroom. So you get to study it in the classroom, and here you get to study it at home. Well, of course, we don't get to meet in the classroom, so this is going to be what you're going to be responsible for. But it is going to be pictures of these models that are going to be on the exam, not the cadaver. So on the exam, I could have a picture like this. And basically, the majority of the test is going to be something like this. An arrow pointing at a muscle and me asking one of 4 questions. Identify the muscle, identify the origin of the muscle, identify the insertion of the muscle, identify the 1 action, 2 action, 4 actions of that muscle. And those are the 4 questions that are on the handout. However, and let's say this exam is usually somewhere around between 80 and 90 questions. 80 and 85 questions is usually where this test ends up, 80 and 85 questions. I guarantee you that somewhere around 10 to 15% of these questions are going to instead be something like this. I have to cheat for a second to do this. So easily 10 or more of the questions on the exam instead will be something like this. You'll see a picture of a bone. You'll see a picture of an arrow. And the question will be, identify the muscle that originates from that bone feature, or identify the muscle that inserts into this bone feature. So I guarantee you bones are going to be on this exam. You are responsible for the origins and insertions. We will be drawing the origins and the insertions. You've learned your bone features already, and there's not going to be any new bone features. They're all bone features you learned in the last part of the class. So you get to use those bone features a second time. I haven't even looked at your lab exams yet, so I don't know how well you did. I'm going to hope you did well on them because you get to use them again. Well, I am a neuroanatomist. One last thing that I guarantee will be on the exam. Obviously, models like the skeleton, like the muscle, the sarcomere, those things that I showed you, and then charts and things along those lines. But there will also be somewhere between, I'm guessing, 2 and 5 questions with something like this, where again, while it's fun to ooh and ah at these bodybuilders, one of the nice things about them, especially when they're nice and dehydrated like this, is we can clearly see the muscles. So I could ask you to identify that muscle, or I could ask you to identify that muscle, or I could ask you to identify this specific muscle or that muscle, right? The advantage of some of these bodybuilders is we can very clearly see the musculature of them. And so you will be responsible for the superficial muscles of the bodybuilder. And in fact, on Canvas, as I'm sure you know, because I'm sure you've looked at it, not only is there this handout for the origins and the insertions, but I'm sure you also noticed that under here, there is a bodybuilder anatomy. Because when you get to be this mutantly big size, there is some minor displacement of muscles from what we see in a typical model. So, so here we can see, why has that been through a bad breakup? How did you guys get on Jay Cutler? What did I miss?
I was like, not another knockoff of the Arnold Schwarzenegger.
Oh, no. But so I guarantee you that there will be somewhere between 2 or 3 pictures from the bodybuilder pictures where you need to identify muscles, superficial muscles in this fashion. But again, we'll go through all of that together. So that's what we're going to be doing in lab. So for lab, you're going to need the origins insertions. You're going to need your study guide and need a red and blue pencil, or pen, so that you can draw those origins and insertions, because that's what you're going to need to be successful on the exam. All right. And I think that's everything. That's going to be this exam. I think those are all the big key things that are going to be on the lab exam. So now you know what to expect for that, and we'll start working on that, and we'll start working on the physiology on Thursday as well. All right. So come caffeinated, come prepared. Thursday, things get fun. And again, I appreciate that this is a lot of material. This can be overwhelming. Welcome to Anatomy and Physiology. I appreciate it's unintuitive. Welcome to Anatomy and Physiology Online. The one nice thing that I will point out to you is this section, we do have spring break right in the middle of it. That doesn't mean you get a week off where you don't have to do anything. That gives you a week of extra studying to master this material and make sure you're going to be successful. So you've got almost a full month. Wait, no, hold on, I'm looking at the wrong one. Over a month. I said over a month to study and learn these muscles. Think of it this way. If you take this over in the summer, you have 14 days basically to learn all the material that you guys are getting a full month to study. So be glad you're taking this now and not in the summer.
What kind of people take it in the summer? I cannot imagine like taking a course like this in the summer.
They're a different breed.
Trying to think of the nice way of saying it. How about, let's say, delusional students?
Yeah, I was gonna say you can't be sane and take a 5-credit course over what, 12 weeks?
It is brutal, and again, there are plenty of class. Look, I never in my entire academic career, graduate and undergraduate, I never took a summer school class. So I've never in my entire academic career taken a summer school class. But what I'd say is that while I see the merit in taking some summer school classes, where a single standalone class, where something where you can kind of cut the corners a little bit, or they can maybe be able to go more superficial, but especially in 430, everything I'm giving you, you need not just for this class, but you need for 431 as well. So some people really like the cram and purge format of the fast pace of a summer class where it's only 8 weeks. But the problem is you need to retain this information, and just cramming and purging doesn't typically help you to retain. So I've actually had students who got an A during 430 during the summer. I've actually had two students who got A's during the summer 430 class and ended up dropping 431 with me during the fall because they didn't remember any of the stuff and they felt really far behind. So I don't recommend it. I spend the first hour of class telling the students not to take it, but many students feel the pressure of time, and so they choose to make that decision. And again, there are some students who get A's, but I would say the majority of students don't get the grades they want, and especially when we're doing it online. You've got so much going against you. It seems kind of brutal. All righty. So there you go. I'll get off my soapbox about that. Any questions on anything else?
When did you say the exam was? I'm sorry.
When did I say what?
The exam was the next one?
April 13th, it looks like.
Okay.
Yep. And again, that's on the syllabus. That's on the schedule. So you have all those things as well. But I got my cheat sheet up in front of me. All righty.
Wait, for the bones, how you gave us a sheet to like memorize of only the bones and bone parts that we need. Do you have the same thing for muscles?
Yes.
Is it that muscles list?
It's that muscles list. And actually, that's a great point. That is one last thing I wanted to say about that muscles list. Let's go back to that one.
So these are all the muscles we have to learn. That's
not only are these the only muscles you have to learn, which if you think about it, remember with other 600 named muscles, it's not too scary. But I want you to notice one other thing about this handout. So notice, for instance, the zygomaticus muscle or the sternocleidomastoid muscle has an origin and an insertion. However, notice down here at the platysma, I've got dotted lines for origin and insertion. Does that mean that the platysma muscle doesn't have an origin and doesn't have an insertion?
No.
No. It just means you don't need to know it. So notice, of all the muscles, all the dozens and dozens of muscles that are in the head and neck region, you are only responsible for 10. And of those 10, only 5 of them do you need to know the origins and the insertions. So for every muscle on the list, you must know its actions. But notice only the muscles that are bold have origins and insertions that you're responsible for. And that's true not just for the head and neck, but notice that's true for the trunk muscles. That's true for the upper extremity. That's true for the lower extremity. You're only responsible for the origins and insertions of the ones that are listed. These that are not listed doesn't mean they don't have them. It just means you're not responsible for them on the exam. All right. So yes. So again, if you want to spend the time looking up what the origin and the insertion of the orbicularis oris is, more power to you, but it won't be on the exam. This handout and the histology, and then of course everything we talk about in lecture, but this and the this handout and the histology is the majority of our lab exam. All right. So yes, thank you. I'm glad you asked that because I want to make sure that that was a point. This doesn't mean, hey, go look it up. No, it means that yes, it exists, but you don't need to know it for the exam. You only need to know the origin and the insertions of the bold muscles, which I'm pretty sure it says up here somewhere. Yeah. Okay. All right. Yeah, so hopefully it's not too scary. Although it'd still be scary. Still be scared, still work hard, because it's still going to take a lot of work. All right. Any other questions?
No, you have a great day, Professor.
I have more grading to do. Unfortunately, like I said, last week I had both your exams and 431 exams, and I am very behind on the exam. So I think someone was just asking that. There is no chance that I'm going to have your exams graded by Thursday. I will get them graded as soon as I can. As soon as I get them graded, I will post the grades. And like I said, when we meet in class, I will then release the exams so that you can observe them, and because I want to make sure that you have the opportunity to look at them. So I will get to them as soon as I can, but it's not going to be this Thursday. I usually try to get things done within a week, but unfortunately with 2 tests in the same week and all the other craziness that's going on, I'm behind on that. So I will do the best I can, get those graded and get those to you. All right. So thank you for that. Any other questions? You guys have a good day, and I will see you on Thursday.
Thank you.
Bye.
Bye.
Thank you.